जी, और खैरियत से हैं आप?
जी जी, सब लोग ठीक हैं, सब ठीक
जी,
है।
बस इंशाल्लाह शुरू ही हो रहा है। अस्सलाम वालेकुम, डॉक्टर नजरुल इस्लाम। हाँ, हाउ आर यू? उमरी साहब।
सर, अस्सलाम वालेकुम।
वालेकुम अस्सलाम, खैरियत से?
सर, हाउ आर यू सर?
जी सर,
शुरू करें?
2 मिनट रह गया सर, 2
Okay.
मिनट सर। या
ठीक ठीक।
So again, a good morning from me now.
Sure, thank you. Yeah, good morning.
So I had some little technical problems, but now
Okay.
I'm completely here.
Okay.
Thank you.
Good morning, Dr. Mohammed. I'm Dr. Shashibhushan here.
Okay. Thank
Good
you.
morning, sir. Good
Yes,
morning.
sir.
Good morning. Good morning.
गुड मॉर्निंग। गुड मॉर्निंग। 1 मिनट लेफ्ट। डॉक्टर साहब टाइप स्विमिंग कर दीजिएगा। 1 2 3। Good morning to all of you. Here, this is Dr. Muhammad Alimuddin Khamri, the organizing secretary of this 2 days international webinar on understanding COVID-19 pandemic and integrated approach. I, on behalf of the organizing committee, the Director National Staff of Unani Medicine, welcome all of you, the panelists. We have I'm very much delighted to invite Professor Syed Ziaur Rahman, Dr. Nazrul Islam, Dr. Claudia Perkel, and Dr. Abdul Wadud. He is my mentor and the director of this institute. Dr. Maria Kahar from Iran, Claudia Perkel from Germany, Dr. Nazrul Islam from Bangladesh, Dr. Shashi Bhushan is one of the renowned pulmonologists of the Bangalore city from Bangalore Medical College and Research Institute, and we are expecting few more. The other panelists were likely to join. As the program is scheduled to start by 11 o'clock, I request our director, Professor Abdul Wadud sir, to hand over to you for the inaugural speech and the welcome address of this international webinar. Over to Wadud sir, please.
Assalamu alaikum. A very good morning to all of you. This is Abdul Wadud, the Director of National Institute of Unani Medicine. Welcome you all on behalf of the Institute, the organizing committee, and myself. The National Institute of Unani Medicine is an autonomous body under Ministry of Ayush, Government of India, situated at Bangalore. This institute is offering postgraduate education in 10 departments and PhD in 2 departments. This institute is a premier institute of India in the field of Unani medicine. in the field of research, education, and patient care through Unani system of medicine. This COVID-19 has posed certain challenges before the medical science to think in a different way. Though scientists, modern medical doctors, Ayush doctors and practitioners have tried their best to tackle this situation, but on the other hand, it has certain challenges to think in a different way about the pandemic diseases. The time has come to come together with an integrated approach. The present seminar brings experts of the various fields together to chart out a way forward to think with a different integrated approach. In the 2-day seminar, we have galaxy of internationally reputed speakers from the field of virology, epidemiology, pulmonology, pharmacology, analytical chemistry, and Unani medicine. These experts are Dr. Ravi from a field of virology, Dr. Sridhar R. Babu, a renowned epidemiologist, Dr. B.L. Shashi Bhushan, pulmonologist, Dr. Mariam Kayat Kashani, an analytical chemist, Dr. Manjunath M. Venkatswamy, a neurovirologist, Dr. Muhammad Kamil, scientist, Professor Syed Ziaur Rahman, renowned pharmacologist and man behind many successful programs of pharmacovigilance, and Dr. Claudia from Germany, a cultural trainer, a cultural trainer and a blogger on Unani medicine. Two of our former students, Dr. Fazina and Dr. Nazrul Islam from Bangladesh and Sri Lanka respectively. It is my privilege to welcome you all once again from this 2-day seminar. In this 2-day seminar, more than 600 delegates have registered. Some of them are also from Bangladesh, Sri Lanka, UAE, and some are from KSA and USA also. The objective of this webinar is understanding of COVID-19 and other pandemics with the integrated approach. I welcome you all once again. Thanks.
Thank you very much, sir. I welcome Professor V. Ravi sir from National Institute of Mental Health and Neurosciences, who just joined during the address of the director of this institute. I am Dr. Mohammad Aliuddin Khamri, the coordinator of this program, sir. It is a responsibility assigned to me to give a keynote address. Otherwise, I can say it's a privilege for me to place some of the key points from as organizer of this webinar, so that to understand the audience and this the panelists. The pandemic COVID-19 affected more than 190 countries and different states all over the world, with more than 3.5 million of the people are affected, and further it is reaching towards 4 million of active cases is supposed to be, where 99% of the cases are mild and only the 1% of the cases are reported to be critical. The global scenario and the current reported condition of the cases reported with the highest number of the pandemic COVID-19. If you look at this globally, United States of America has occupied highest position with more than 7.6 million cases, and unfortunately, India has been occupied the second place, keeping Brazil side with more than 6.6 million of cases, followed by Brazil more than 5 millions, Russia 1.2 millions, and Colombia 86 lakhs. And the global fatality rate of this COVID-19 is around 2.95%, and the national fatality rate, according to Indian statistics, is around 1.6%. To the best of our understanding, though this is a novel disease, the etiology of the COVID-19 was clear that it is a novel coronavirus. That is one of the species of the SARS-CoV-2. First case was reported in Wuhan, China, sometime in December 2019. That is the name is given as a COVID-19. The first reported case was in December 2019, and some of the historical reviewers says that the cases are there before December 2019. Somewhere it was August 2019 only the cases are reported. In India, reported the first case on 30th of January 2020, that's 2019, where the infected person was reported from Kerala with a travel history of China. Globally, we understood that the disease is transmitted by means of the contact, a direct contact with the infected person, of the fomitic infection or aerosol inhalation of the organisms. It depends upon the viral load, the severity of the disease depends upon the viral load, how much that people are being infected. Symptoms also, there is a wide range of symptoms have been reported, starting from fever, tiredness, breathlessness, gastrointestinal symptoms, several things are there, including loss of sense and taste, everything was there. Finally, the modern science came into the stream. They have tried to find out the diagnostic techniques through which 70% of the cases were found to be positive with the high resolution CT thorax. It gives a more confirmation about the disease, as because the major symptoms of the disease are reported as in the form of a viral pneumonia. That is the reason 70% of the cases were found with the viral pneumonia like symptoms in this high resolution CT. And other investigations like digital X-ray may not be proved as a diagnostic tool to find out the investigation. Some of the investigations like rapid antigen test is a big question mark with the confirmation of the disease, and RT-PCR has occupied a very major role in confirmation and the diagnosis of the disease and that infection. Apart from that, there are several organizations, several agencies, several scientists. They have tried to find out different antibodies related to this infection has also been found. Right now, the trend is going with the RT-PCR as one of the primary investigation tool through which they are they are detecting the infection. As I told you, that's because that mortality is also reported around 2.82% globally, and India is evidence of 1.6%. National fatality is about 1.
Sir, you have muted yourself. Dr. Mohammed?
Yes.
Yeah, you're muted. Your voice is muted.
Is it audible?
Yeah. Go ahead, please.
So in the form of a new hope with reference to the medicines which were intended to have an efficacy in controlling COVID-19 are something the monoclonal antibodies, recombinant AC2 inhibitors, and interferons, ivermectin. These are the drugs where the people are expecting some new hopes with this modern drugs. Some of the promising evidences are also there with some drugs like the dexamethasone, remdesivir, convalescent plasma therapy, where there are difference of opinion. Most of the observations are there. Convalescent plasma therapy is having a mixed kind of responses. And the mixed evidence of positive use of the medicines are associated with the favipiravir and tocilizumab. These are the two drugs where the people are thinking of mixed response. There are very few minor evidences are reported with antiviral drugs like lopinavir, ritonavir, and hydroxychloroquine. It was promoted as a preventive prophylaxis medicine and also therapeutics. As a whole, the modern science, the conventional medicine, people are effortlessly trying to bring some medications to control the disease, and the prophylaxis also been most important. Overall, the disease was spread globally, and there was a huge spike of a death was reported in the month of April 2020. And if you look at this for the last 17th August to 22nd September, the survey shows that India is also reaching to the level of United States with reference to the prevalence of the disease around 9.3%, and followed by Brazil 2.8%, Spain 4.6%. Moreover, if you look at India, there are certain states where the highest number of cases are reported, where Maharashtra has occupied the highest cases, And now with reference to that mortality rate, Punjab has reported 3.05%, followed by Maharashtra 2.64% and Gujarat 2.44%, Delhi 1.89%. Luckily, the people who are from the Karnataka, it is 1.45%. I'm very happy to say Kerala is the only state which is reported 0.37% of the national mortality rate. But since for the last 48 hours, the report says that there is a huge surge, a spike of infection in Kerala, Chandigarh, Pondicherry, some of the places where the infection, probably the second wave of infection is going on. So it has become a biggest challenge to the global scientists, that people who are working for developing of the treatment. Now, the point here to be noted here, it is being cured, no guarantee. Whatever the drugs are being used for the management of this infection, even though there are cases reported of the reinfection. In India, we have reported cases in Indore, Bhopal, even Karnataka also there are cases. After 21 to 30 days, the reinfection was there. Probably the reason it could be hypothesizing that several reasons for the reoccurrence of infection is there. Even the people who are treated, RT-PCR and other investigation shows negative, but reoccurrence of the infection is probably hypothesized that there may be a low immunity of the individuals, or falling levels of the antibodies are also being reported, because antibodies the one they are going to prevent further infection. Here the within 3 months and 90 days there is a natural declining in the number of the antibodies also been reported. Ultimately the people are looking forward for developing vaccines. That is the only hope to prevent and to manage the condition. According to the some of the information from the global scenario, around 42 manufacturing units are agencies which are thoroughly working for development of the vaccines, and the reputed firms like in India, we have Serum Institute of India, Pune. They are going to have a vaccine candidate CDX005, that is Codagenix, and Bharati Biotech from Hyderabad, Covaxin, AstraZeneca and Oxford, COVID Shield. Like there are so many that Innova, Moderna, Pfizer, CureVac, Institute, Pasteur, Med, Themis, AstraZeneca, like around 40 agencies are they are working for coming out of the vaccine development. Coming to the my science, the Unani system of medicine, which speaks about the science. Unani system of medicine is a very classical science. The science, the philosophy, or the science is based upon the theories and the principles of Hippocrates' body fluids, to whom we call as the theory of humors or akhlaq, theory of Nazari akhlaq, where 4 different body fluids are there: blood, phlegm, yellow bile, and black bile. The quantitative and the qualitative equilibrium of all these 4 body fluids will maintain health. Any disequilibrium leads to diseases. Apart from that, Unani speaks about the importance of tabiyat, that is physis, medicatrix naturae. There's a Greek classical physiology, Greek science says that there is a term medicatrix naturae, which is called as something like it stands for the physis, means the tabiyat, which take care of all the human body's regulation of disease and health. Unani system also pays very much importance to the asbab sitta zaruriya. There are 6 essential factors which are mandatory for all the living beings, because without which nobody can survive. These 6 essential factors, they are dormitorily associated with the individual throughout the life, without which no living being can be survived. Like water, what we eat and what we drink, the physical rest and repose, psychic rest and movement, followed by sleep and wakefulness, elimination and excretion. These are all the 6 essential factors. The modulation of these 6 factors will keep the people healthy as well as prevent some of the infectious condition. Unani system of medicine, it is a old time-tested system of medicine with a record of more than thousands of years, where we have a clear description about the diseases. It includes infectious disease under the chapter of Amraz Waba. Waba stands for infection, Amraz stands for the diseases. Under our literature, that clearly they have explained about that the authorities like Abu Ali Sina, Razi, Ibn Zuhr, Ibn Rushd, Razi, as I told you, Rabban Tabri, and many more authorities, they have clearly explained about the pathology, the relative organisms. Also, they had the concept, they not only believed about the imbalance of these 4 body fluids. Apart from that, they have equally paid attention toward the ajazamic kabisa, Mean they were thinking about there are certain organisms which are responsible for the cause of the disease that causes disequilibrium of the body fluids also. Here in our literature, though the COVID-19, Zika, Ebola, these are all the new names of the viral infections, but we have some of the relatively similar kind of a diseases description, and those diseases are treated by our ancient Greek physicians. Even the modern Unani practitioners are also treating all such conditions. We had a good history of treating smallpox, measles. I would like to emphasize upon the work, the contribution of Razi, whose masterpiece on particularly, he is the person who differentiated smallpox from measles clinically, and he came out of the compendium Al-Judri wal-Hasba, a masterpiece on which he clearly explained about the viral infections, particularly the smallpox and the measles, along with how to prevent this infection, the concept of epidemics, concept of quarantine, and indirectly he was explained about the development of natural immunities also there in that book. We had a history of understanding smallpox, measles, Hamirasaifi, cholera, and more particularly a wide description on influenza is there. Today, the COVID-19 and the recent past, the other viral infections, what we have reported are more or less a form of influenza only. Those classical literature, they understood well influenza, they managed those diseases at that time with the existing indigenous sources of medication, but they have framed in clearly how to manage all such infectious conditions, particularly influenza. According to Unani system of medicine, these infectious diseases are basically causes. There are 4 different etiological factors which are responsible for the manifestation of infectious disease, where they have paid more emphasis upon the environmental factor, where the changes in the air, water, earth, and certain influence of the celestial bodies also being paid attention by the Unani Tibba. If you know that as long as the water and air are pure, they are not going to do any harm to the pure living beings. If any substance, organism, to which they have called it as ajazam khabisa, ajazam means organisms, khabisa means that are vitiated. They have the tendency to do harm to the living beings. Mean those days they understood about the importance of the body fluids, but at the same time they are incited to understand the microorganism. But because of they don't have the devices to understand the structure and the mechanism of actions of the organism, they understood that there are certain organisms which are also causes derangement in the body fluids. Like for every infection, Unanis believe that there is a change or the certain factors should be available, like hotness, and there's hararat, rutubat, and organisms are very mandatory. All such conditions were treated with different drugs and regimens are also there. I would like to emphasize some of the regimens, like the principle of Unani says that if the disease is of a particular temperament, mostly influenza and other diseases are of cold in temperament. All such diseases were treated with the contradictory to the conditions with the hot temperament. The temperament is nothing but, according to Unani, we call it as a mizaj. If the disease of the mizaj is barid, cold, then regimens and the drug should be used contradictory to that one, and all the things should be used hot. So we have a list of the drugs. We have a list of regimens, like there are aromatic therapy where use a number of hot oil vapors, buhur, that means giving fumigation kind of things. There are zimats, there are lakhlakhat, there are nuzuhat, there are nufukhat, there are nushuhat, and there are procedures of giving fumigations also. Apart from that, the drugs are there. Most of the drugs which contains mur, zafran, elwa are used as a prophylaxis as well as the therapeutic medication during the period of epidemiology. And particularly they have paid more importance about the air purification system. And for that, they use some of the volatile substances. They keep the volatile aromatic substances in the windows. So the air should come, the flow of the air should be pure and prevent from the microorganisms. There are so many formulations which is dealt in the Unani system of medicine, like kurse kafur, kurse bedamushk, khamire gauzaban, khamire marawarid. And recently the government of India during the pandemic, they have purported particularly the Bedana, Unnab and Sipistan along with the Khamire Marwarid as best prophylactic medication to control secondary infection as well as to prevent the further complications of the coronavirus. And alternative system of medicine, they paid much emphasis about providing joshandas or quarts, that is nothing but the decoctions, where mostly they have used some of the traditional spices as decoction, where they use tulsi, darchini, karanful, filfiladeras. All these things have been, and most of the people, they got response also. Unani has paid a much very importance on the use of antidotes that's called as a theriacs during the period of infection, where theriac babai, theriac afai, theriac arba, like there are many more theriacs are there. They have a specific activity against the microorganisms, and mostly even nowadays we are using to prevent further infection as a prophylaxis as well as therapeutics along with adjuvant therapy during the pandemics. With these few words, and there are many more things which is coming out, but as I believe that this COVID-19, it is a big challenge to globally. Still it is in the phase of understanding the organism. There are a lot of conflicts and the controversies with that. Till we don't have any clear drugs, we are in the process of developing vaccines. All alternative system of medicines, they are just trying to provide the immune boosters, because alternative science believe that once the immunity is strong and it can fight against the disease, it provides strength to the body. That is the reason almost all alternative system of medicine, including Unani, is also propagating frequent use of all immune boosters like Khavere Varvaris, as I told you, Bedana Unnapsipistan, antidotes are all there, and they are helping. But at the same time, I believe that it is only possible with an adjuvant of modern sciences and other sciences and the development. We can together work, we can together fight against the disease. Till today, I believe that only social distancing, frequent hand wash hygiene, masks, they are playing very vital role in prevention of the disease, and the rest of the medication from the conventional system of medicine and alternative system of medicine globally, they are still struggling to find a correct line of treatment for successful management of COVID-19. Thank you. I take a privilege. We have our speaker, the first speaker to whom we have invited for this international webinar. I'm very privileged to invite Dr. V. Ravi sir, who had completed his MBBS from Madras Medical College and MD Microbiology 1983 JIPMER Pondicherry, and he's the one pioneered who started the Department of Neurovirology at National Institute of Mental Health and Neurosciences, Bangalore in January 1985. Sir presently working as a senior professor and head of the Department of Neurovirology at NIMHANS. His areas of research includes public health virology, outbreak investigation. Sir is doing a lot of work nowadays. He's associated with this pandemic control in Karnataka. He's one of the member of the task force committee. He developed indigenous diagnostics, acute encephalitis syndrome, biology, pathogenesis and immune response to Japanese encephalitis, chikungunya dengue, SSPV, HIV and neuro-AIDS, immunology of the psychiatric diseases. Sir has received 25 research grants from the national and international level reputed agencies like ICMR, Department of Biotechnology, Department of Science, DST, CSR, internationally from Indo-USA Vaccine Action Program, National Institute of Health, CDC, USA and MRC, University of Liverpool, National Institute of Health Research, UK. These are all the very few funding agencies from which Sir has taken a lot of research projects and working. Till today, Sir will be having more than 160 papers. Sir has contributed 25 chapters in the books and monographs, compiled and edited, author 4 WHO monographs in virology. He is an authority in the virology. Sir has right now holding one European patent on Cyticercus cellulosa diagnostic development. This patent has made in collaboration with the Exciton Diagnostic Private Limited, Bangalore, developed 3 commercial ELISA assays for HIV, HCV, and JEV. Sir monitored more than 25 PhD students as a guide and co-guide, and he guided several MD psychiatry, MD neuro students. Sir has received fellowships from USA 1992, Commonwealth Medical Fellowship, UK from 1995 to 96, WHO Short-Term Professional, SERA 2006-7, International Examiner for MD Virology Program of MRI, Sri Lanka since 2007, Visiting Professor Microbiology and Immunology for University of Liverpool, UK since 2013. Presently, as I told you, Sir is the task force member of the Karnataka COVID-19, and he is the member of the ICMR mentor for COVID-19 testing labs. Sir served as an expert member for various task force committees and scientific advisory committees of WHO, DBT, DST, ICMR, NIV, NARI, etc. I'm very happy to place before you. Sir has been awarded with the C.V. Raman Young Scientist Award of Government of Karnataka in 2000, Dr. J.B. Srivastava Oration Award for ICMR, as an eminent medical virologist 1997, Dr. C.S. Agrawal Oration Award of IAMM 2008, Eminent Medical Virologist Award of Indian Virology Society in 2013, elected Fellow of National Academy of Medical Sciences 2009, and Fellow of Indian Academy of Sciences 2010, Dr. H.I. Chala Award of in 2016. Sir already served the Indian Association of Medical Microbiologists for 10 years in various capacities, and he's a member, EC member from 2002, Joint Secretary, President for different organizations, and very interesting to know, Sir is a master of virologist, but at the same time, Sir is also having some of the habits like cooking, conducting quizzes, and he's also writing rhymes. With this brief introduction, it's over to Dr. Ravi Sir, head of the professor, senior professor, virologist from National Institute of Mental Health and Neurosciences for your presentation.
Thank you, Professor Alimuddin, for the invitation and the kind introduction. I would like to express my deep sense of gratitude. It's an honor to be a part of this international seminar that you're holding. I bring felicitations and greetings to all the fellow panelists whom I see on the screen. What I will do in the next 20-25 minutes is to share with you some aspects of, can you see the screen, sir? Can you see my screen?
Sir, please share your screen from your end, sir. We can able to see only that your presentation.
Can you see it now?
No, sir.
1 minute.
Please.
So I will be talking to you on SARS-CoV-2, which is virology of SARS-CoV-2. How do I share this now? Okay. Can you see my screen now? No?
No, sir. You please share that, sir. No.
Yeah.
Now, sir, we can able to see, sir. Wow.
Thank you. I'm sorry. I'm technologically challenged.
No, sir. It's okay, sir.
Okay. I'll be talking to you on virology of SARS-CoV-2. This is the outline of my presentation. I'll give you a brief introduction, the structure, genomic organization, physical chemical properties, biology of the virus, replication, which is very important from various points of view in understanding pathogenesis and developing therapeutics, some aspects of pathogenesis, and summarize with what are the current therapeutic approaches from the virus cell biology point of view and the vaccine approaches. What do we know about coronaviruses? Coronaviruses have been around for 50, 60 years. The name corona comes from the fact that it resembles the crown. A crown with, which is spherical in shape and with projections studded on all the sides of the virus. Because it resembles a crown, it's called corona. These are basically enveloped viruses. They have RNA as a genetic material. Prior to the SARS-CoV-2 or the novel coronavirus in December 2019, there were 6 coronaviruses which are known, 4 which have various numbers as shown in the slide, 229E, OC43, NL63, and so on. All of them were known to cause mild flu-like symptoms, and some of them cause gastroenteritis. It was in the turn of this century that the first severe acute respiratory syndrome coronavirus was discovered from southern part of China again. It spread globally to many countries. Fortunately, India was spared. That was in 2002 and 2003, and it died down due to various control measures. Again, in 2012, we had the Middle East respiratory syndrome, or the MERS coronavirus, which was discovered. Coronaviruses are widely distributed across the globe. They exhibit certain amount of genetic diversity. They're found in humans, animals, and birds, and they spill over from one species to another. This is the summary of the reservoirs of coronavirus in nature. There are 2 reservoirs. One is bats and the other is rodents. Rodents have coronaviruses which cause infections primarily in cattle. The bat coronaviruses are reservoirs from where there's a spillover that occurred to human beings. For example, the SARS coronavirus, the original SARS coronavirus 1 was a spillover from pigs. The MERS coronavirus was spillover from bats to camels and then humans. And the recent SARS-CoV-2 is a spillover from bats to pangolin, which is an animal belonging to the anteater family. And these viruses probably spilled over from pangolin to human beings in a wet market in Wuhan, which Professor Alimuddin referred to in the late part of December of 2019. And this is called severe acute respiratory syndrome coronavirus 2 to distinguish it from the SARS-CoV-1. COVID-19 is a name coined by WHO in February. It's an acronym for coronavirus-induced disease. 19 is because it was discovered in 2019. And SARS-CoV-2 is the name of the virus that causes the disease. If you look at the structure of coronaviruses, they're fairly large viruses. They can measure anywhere upwards of 60 to 150 nanometers in diameter. They're spherical. On their surface, there is a very prominent protein which gives the crown-like appearance. These are projecting on the surface of the virus. And this protein is called the spike protein or the S protein. Very, very important protein because the virus attaches to human and animal cells using this protein. So this basically provides the key to unlock cells for the virus to enter. This is embedded in an envelope, which is depicted in the cartoon on left as a blue circle. Also embedded in the envelope is a membrane protein and an envelope protein. Each of them have different functions. This envelope shields a core, a protein core, which is made up of by the nucleocapsid protein, and inside the nucleocapsid protein is a single strand of double-stranded ribonucleic acid, which is the genetic material. This is the electron microscopic picture of SARS-CoV-2, and you can see this is from National Institute of Virology, where they isolated the virus in February 2020. If you have a closer look at the spike protein, the structure is shown on the left. It has been crystallized. It's a trimeric protein which has a head which is globular and a stem which is linear. Embedded in the head is a very unique loop known as the receptor binding domain. This is unique to SARS-CoV-2. It distinguishes it from SARS-CoV-1, and this is one of the unique proteins of the virus. When the virus multiplies in cells, it also makes a lot of other proteins, which are raw materials for assembly of the virus. Now, these are called non-structural proteins because they don't go into the final structure, but without these proteins, you cannot make viral particles. There are a number of non-structural proteins, 16 to be precise. I've shown it in a tabular form on the right. The reason I wanted to touch on non-structural proteins is several of them have very profound effect on the human immune response, which is very relevant for pathogenesis. The viral genetic material itself is a double-stranded RNA of positive polarity. What do you mean by positive polarity? When you inject this RNA into a cell, it can directly go into translation of proteins. Negative stranded viruses cannot do that. When you have a negative strand virus like influenza, you inject the virus, first it will make a copy, much like the old photographic negative from which you make a print. Here, this is directly translated into proteins. That's why it's called positive polarity. It measures approximately 29,000 nucleotides. It has many genes, starting from non-structural proteins to structural proteins. Very interestingly, the initial proteins that are translated from an open reading frame 1 called ORF1A and ORF1B are proteins that are immediately essential for translation of virus genetic material. If you look at the phylogeny of this virus, the SARS-CoV-2 is depicted here in the center as 2019 novel coronavirus. You can see it is very closely related to the bat coronaviruses. So there have been several speculations that it is a laboratory engineered virus. I won't go into the controversy, but as of now, there is no evidence to say it is manipulated in the laboratory. Usually when you manipulate a virus, there are telltale evidence left behind in some form or the other. This virus has 99% similarity to the pangolin virus and 98% similarity to the bat viruses. So this is, as of now, many people agree that it is a natural virus. It's not an engineered virus. Coronaviruses, fortunately, are very stable. They don't mutate as much as influenza viruses. Nevertheless, being an RNA virus, whenever it replicates, it does make errors in copying. So RNA viruses are poor photocopying machines. When they copy, they make some errors. So also the SARS-CoV-2 makes mistakes, and this is exploited to type the virus into various clades. Today we have around 6 clades known of coronaviruses, and this is the global distribution. They're designated as A, 19A, 19B, 20A, 20B, and 20C. Now they are distributed widely across the globe. Different areas have different viruses, but the parent virus for reference is the Wuhan virus. Now this is the phylogeny depicted in a graphical form. We have done sequencing of close to 100 genomes, full-length genomes from clinical samples in our laboratory, and we have found all 6 clades, predominantly A, B.1.1, B.1, B4, and B6 in our state. Now these clades we've been able to associate with clusters. A cluster is a group of cases where it has spread from one index patient. Now we have various clusters. You can see a table below in different colors. They depict the different clades, how the clusters have progressed. We have a unique cluster which is purple on the top here. This is the Nanjangud cluster, a cluster of patients that occurred in a pharmaceutical company in Nanjangud close to Mysore. This is a unique cluster. It is a cluster that has burnt itself out. No more spread is occurring from this cluster. The other clusters that you see is the orange one is the predominant one, B.6, and various variants of it. Now this is the largest cluster. This was introduced into Karnataka, into various parts of Karnataka by travelers, mostly domestic travelers who attended a congregation in Delhi. So these are the two major clusters that we have seen. And on the right, you'll see a cluster analysis showing beautifully how an index patient led to spread to a large number of others, from them secondary, tertiary, and quaternary spread. So that is the power of the molecular epidemiology, that you can track viruses, how they are spreading. The physical and chemical properties of SARS-CoV, it is a thermolabile virus. It is inactivated at 70 degrees. When you expose the virus for 5 minutes, it is killed by boiling water. Chemical stability, because it's an enveloped virus, envelopes are made from host lipid. All lipid solvents like ethanol will dissolve the lipid, the spike protein will fall off, and it inactivates the virus. Other routine disinfectants based on chlorine or peroxyacetic acid also disinfect the virus. Soap is the best disinfectant because it dissolves the lipid coat of the virus. So washing the hands with soap is very, very important. It's much more cheaper than using a sanitizer. So I would recommend hand washing with soap and water, which is very good. So coronaviruses are known to survive on various surfaces. It depends on whether the surface is porous or non-porous. On porous surfaces, it can survive for about 2 to 3 hours. On non-porous surfaces such as steel, it can survive for 6 to 8 hours, after which, due to the environmental conditions, it dries up and gets inactivated. So one of the major mechanisms of preventing transmission is to use protective equipment like masks. Triple layered masks are effective, but not effective as N95. N95 in N stands for 95% protection. It filters out various droplets less than 3 microns, above 3 microns in size. And because the virus is transmitted through droplet infection, if you can prevent droplet from entering, then you can prevent virus infection. How does coronavirus enter and replicate in human cells? We know that coronaviruses, SARS-CoV-2 especially, uses a very normal physiological molecule present on human cells known as angiotensin converting enzyme 2, or ACE2 receptors for short. The virus spike protein specifically locks into this protein, and once it locks in, there is a transmembrane serine protease that clips the spike protein and helps in fusion of the virus envelope with the cell envelope, and then the virus is delivered inside. This is one of the mechanisms. There's another mechanism I'll talk to you later. Now, ACE2 is the receptor. It is distributed widely across various cells in the human body. Highest density of ACE2 receptors are found in the human gut. It is also found at very high levels of expression in human testis and the human kidney. Moderate level of expression is found in the liver, the lungs, the heart, the blood vessels, and even expression of ACE2 receptors is found in the brain, in neurons. So as you can see, all these cells can be targets for coronavirus replication. What actually happens is the virus attaches to ACE2 receptor, the membranes fuse and the virus is delivered inside. Cellular virus enzymes destroy the virus outer coat, the nucleoplasmid, and the viral RNA is released. It immediately attaches to ribosomes and goes into a translation mode. There is a very important gene known as RNA-dependent RNA polymerase that kicks off the initial reaction, making copies of virus genetic material. This is then transported to the endoplasmic reticulum, where being a positive stranded virus, it is immediately translated into various viral proteins. Some of them are enzymes. Viral proteins, large proteins are chopped, they are assembled, they are taken to the outer surface of the cell, and they bud out of the cell. Another mechanism that has also been noticed is not entry through spike protein, but forming a vesicle on the cell membrane, and this inside this vesicle, enzymes digest the virus. Now, this is a very important alternative mechanism, and I will come to it when we talk about therapeutic targets. So people always traditionally in virology try to target virus attachment, virus fusion, virus translation when the genetic material, and then virus assembly and virus release. So the various drugs which Dr. Sashibhushan will talk to you, I will come to the end of the talk, I'll tell you where various drugs act. The SARS-CoV-2 is widely permissive to a number of cell lines of various origin. We know that this virus in the laboratory multiplies in a large gamut of cells, whether it's kidney epithelial cells, lung epithelial cells, human hepatoma cells, human neuronal cells, animal neuronal cells. So in a variety of cells which express ACE2, the virus replicates. And it's a virus that replicates very fast. This graph tells you here at 0 top point when you infect, the first virus production is seen around 6 hours. It reaches a peak usually between 24 to 36 hours and then plateaus off. So it's a virus in 48 hours can reach 6 logs in cell lines. 6 logs means 6 lakh particles, 10 lakh particles in 2 to 3 days, one virus can produce. So that is the replicative ability of this virus. Normally, we titrate viruses by looking at cytopathic effect, which you can see in the panel below. This is Vero E6 cell line infected, and the bottom is the control. You can see within a matter of 24 to 48 hours, most of the cells are destroyed. They clump, they shrivel, they become rounded, and they get detached. And we titrate viruses by doing what is known as a plaque unit, a plaque forming technique. And you can each area of clearing here you see in the plate is a plaque. That means one viral particle which entered and in 3 days multiplied and lysed the cells, producing an area of clearing. It produces fairly large plaques, as you can see, visible to the naked eye. Now, this is a very interesting paper. Those of you who are interested in virology, in Lancet Microbe, published in April this year, showing that the replicative ability of the virus, irrespective of the various species of animals and the cell lines used, whether it's fibroblasts or whether it's kidney cells, virus multiplies very efficiently in all cell lines. Moving on to the pathogenesis of COVID, a very, very interesting virus teaching us many, many lessons. The primary event is attachment to ACE2 receptor. So it can produce disturbances, physiological disturbances related to the enzymes that control blood pressure, such as angiotensin, renin, and the aldosterone system. Because it occupies the receptor, these hormones are not able to bind, so blood pressure can be altered. The virus multiplies and lyses a cell, which we call direct cytotoxic effect. The third mechanism is virus replicates in blood vessel lining of endothelial cells, damages them, And when it damages the endothelial cell, it forms a nidus for platelets to come and attach, and the whole clotting process starts. So microclots can form in the blood vessel cells. Lastly, and probably the most important mechanism is the virus induces a very aggressive immune response in some people, and it is the immune response that actually is responsible for the disease. I've been an advocate for viruses the last 25 years. Don't blame the poor virus. It is the host who decides whether they will get severe disease, mild disease, or moderate disease. Louis Pasteur, father of microbiology, on his deathbed made a very profound statement. When he was dying, he said, "The seed is nothing, the soil is everything." Because you people belong to alternative systems of medicine and you believe in this concept, I wanted to make this point that the seed is nothing, the soil is everything. There are two types of immune responses in allopathy that we talk about. One is the innate, meaning the first line of defense. It is present on all surfaces. It is present at all times. It is the most immediate protective mechanism. Unfortunately, it is not specific and it does not have memory. Whoever comes into the body, it will try to prevent. The adaptive immune response, on the other hand, is very specific. It has memory and it uses a variety of mechanisms to eliminate virus. A lot of things that we have learned in the last 6 months is that innate responses are very crucial, very important. Those whose innate responses are not good, they land up into trouble because the macrophages overreact and produce a number of chemicals known as chemokines and cytokines, which actually promote a great degree of inflammation, whether it's the lung or whether it's the blood vessel. These responses are very crucial. Do we have any evidence to say that? A very beautiful paper published sometime around May, June of this year, showing that innate responses hold the key to severe disease. What did they do? They collected lung fluids from people who are mild, moderate, and severe of proven COVID patients. They also collected mild, moderate, severe other respiratory infections like flu, fluids, and they subjected to whole genome sequencing. And what did they find? You see on the right, depicted as a heat map. Red means very robust, brisk response. Gray means very poor response. In patients with SARS-CoV-2, the bronchoalveolar fluid showed enormous response to a number of cytokine genes, which are expressed by the innate system. On the contrary, other bacterial and viral infections did not show this. So this is a very important evidence to say that the innate immune response holds a key. I will not go into clinical manifestations. Dr. Sashi Bhushan is an expert. He will talk to you. As Dr. Alimuddin said in the keynote address, it's a spectrum of illness from mild to moderate to severe. Fortunately, mild and moderate account for over 95%. The acute illness lasts for 10 days. It can involve various systems. I'm sure Dr. Sashi Bhushan will touch on this. Today, the mantra is, if you know COVID, you know medicine, because almost every system is involved in COVID. So one has to remember the kinetics in SARS-CoV-2 infection. Actually, virus replication occurs very briskly in 1 or 2 days before onset of symptoms. So people can be infectious 2 days before onset of symptoms. The virus levels peak on the day of onset of symptoms, and virus levels in various, especially in the respiratory system, usually wanes by between the 10th and 14th day. So this is the virus peak, 1 or 2 days before onset of symptoms, up to 2 weeks after onset of symptoms. When we say virus, it can be whole virus, it can be virus protein, or it can be virus genetic material. Unfortunately, unlike other infections, virus genetic material shedding can occur up to 6 weeks. Why I'm making this point is, as Dr. Alimuddin said, the gold standard for diagnosis today is RT-PCR, where we detect the viral RNA. Viral RNA can be present in an infected individual for up to 6, sometimes even 8 weeks, I've seen. So detection of RNA is evidence of infection, and it's evidence of current infection. But it is not equivalent to infectivity. Infectivity requires whole virus. So what is more representative of whole virus is probably isolating virus by cell culture, which is not possible. You need BSL-3 facility. So the surrogate is viral antigen, which is nothing but viral proteins that we can detect in. So these are the methods that are used for diagnosis of SARS-CoV-2. Direct evidence is demonstrating viral genetic material through RT-PCR or antigen. Indirect evidence is by showing antibodies. It takes up to 2 weeks for antibodies to develop in SARS-CoV. It's a very, very interesting virus. Most RNA viruses produce antibodies in 2-3 days and reach a peak by 10 days. This delays antibody response. We do not know why. So currently people are they have mapped how many days RT-PCR will be positive. On the graph in the right, you can see for up to 10 to 14 days, then levels of RNA go down. Interestingly, antibody starts picking up on 5th, 6th day, reaches a peak around the 14th, 15th day. So there is a crossover point where both will be positive, and people are trying to see if intelligently we can use a combination of various methods for diagnosis of SARS-CoV-2. Antigen detection test is a rapid test. You get results in 30 minutes. It's a point of care test, much like the pregnancy test. You can see red or pink lines in the device. The advantages being in half an hour, you'll get the result. The disadvantages being it's only 50% sensitive. But the major advantage from public health point of view is if somebody is detected positive in half an hour, they can immediately be isolated and further spread prevented, whereas an RT-PCR will take 24 to 48 hours, during which time more people get infected. Antigen test, the government is advocating for various situations like containment zones, for emergency use in hospitals, and also for contacts, especially if they are elderly and comorbid. Antibody testing is not for diagnostic purpose, but it's very good to know what proportion of the population is already exposed to the virus without their knowledge. We can do sero surveys. We are just about completed the first large-scale sero survey in Karnataka, and the results will be out very soon. Very interesting results. I'll wait for the government to announce it. Finally, let me end on 2 small points. One is therapeutic approaches. What are the approaches you can use? Before the virus enters, you can neutralize the virus by using antibodies. This is the basis of plasma therapy, which Dr. Alimuddin referred to in his keynote. We are trying to prevent virus attaching because a recovered patient will have antibodies that can neutralize the virus, so you prevent attachment. Secondly, you have attachment inhibitors. You can use molecules or monoclonal antibodies, or you can use chemicals that resemble the viral spike protein. These are competitive inhibitors. They compete with the virus for ACE2 protein. Then you can use the TRIM protein, which is very key protein to internalize the virus, inhibitors to that. You can also use various molecules that can act at various stages of virus replication. Translation is usually prevented by lopinavir and ritonavir. These are HIV drugs that are used. Then processing of viral proteins assembly, which remdesivir can take place, and remdesivir, favipiravir, ribavirin, all RNA virus drugs borrowed from other viruses like Ebola and flu and non-specific like ribavirin. You can also use monoclonal antibodies to inhibit cytokine production, especially IL-6. Tocilizumab is a IL-6 inhibitor. So there are various approaches people are taking because we are very desperate to save the life of a person who has a severe infection. There are 184 vaccine candidates currently in various stages of development. The most promising ones that have entered phase 3 trials are recombinant protein vaccines, vectored vaccines, and RNA vaccines, naked RNA vaccines. India has its own 2 vaccine candidates that are progressing very fast. One is the Bharat Biotech inactivated vaccine, and the other is the Zydus Cadila vaccine that is going on. Apart from that, Indian companies are engaged with multinational agencies for the Oxford vaccine trial, which is a backbone of adenovirus, in which a chimpanzee adenovirus in which SARS-CoV-2 spike protein has been expressed. And you also have the Sputnik vaccine from Russia, all of them on various stages of trials. Currently, phase 3 trials are starting. Phase 3 trials usually take 3 to 4 months before license is given, and then you require mass production and then distribution. So we are not talking of vaccine availability before March. Whatever you read in the press that tomorrow you're going to get a vaccine is not true. Believe me, it takes anywhere between 6 to 9 months from the time a vaccine is first used in animals to take it to human administration. I would like to end here and thank Dr. Alimuddin and this great institution for giving me this opportunity. Thank you very much.
Thank you very much, sir. Thank you. There's only one question with you, sir. As basically most of the patients who are tested, rapid antigen test is negative, followed by RT-PCR is also negative. Then there are certain symptoms, mild to moderate symptoms are there. When such patients were subjected to HRCT, they found there are certain ground glass opacity was there. Then how you are going to say this is the reliability of the investigation tools is the biggest one of the question.
Very nice question, Dr. Alimuddin. There is no disease and no test that is 100% sensitive and 100% specific. This is reality. The current PCR sensitivities are between 90 to 95%. Antigen sensitivities are 50 to 80%. Let's take it as 50%. The government of India has given a very clear guideline. If somebody is symptomatic and rapid antigen negative, it is mandatory you have to do an RT-PCR on the same patient. Whereas asymptomatic and rapid antigen negative, the government guideline is very clear, no need for confirmation by RT-PCR. You raised a very important point, which I'm sure Dr. Sashi Bhushan will touch. He's an expert. Virology tests negative still can be SARS-CoV-2. There are very clear-cut guidelines based on clinical manifestations and imaging studies, which I'm sure Dr. Sashi Bhushan will touch on. He has developed guidelines for the state on antigen and nucleic acid negative SARS-CoV-2. It's a reality. There is a proportion of patients who have everything except the test being positive. So it is a reality.
Thank you, sir. Thank you. Thank you for joining with us. We have another speaker. I'm very delighted to invite him, Dr. Giridhar Babu. Giridhar R. Babu is a professor and head life course epidemiology at Public Health Foundation of India. Presently, he is in Bangalore only. Sir has completed his MBBS from Kasturba Medical College, Manipal, and Master of Public Health and PhD from University of California, Los Angeles. He has worked for 2 decades in public health research, practice, and academics. He began his career at a Center for Community Medicine of All India Institute of Medical Sciences. Efforts in stopping polio transmission in the state of Karnataka. He initiated advocacy for measles surveillance in Karnataka, leading to constitution of multi-year plan for measles elimination in India. He was awarded the prestigious intermediate fellowship of Wellcome Trust DBT India Alliance to start a cohort study. The cohort is named as MAASTH, M-A-A-S-T-H, MAASTH, that stand for Maternal Antecedents of Adiposity, studying the transgenerational role of hyperglycemia and insulin. Sir is having more than 80 papers published in the national and international high impact journals. He is the member of Epidemiology and Surveillance Research Group constituted by Indian Council for Medical Research, National Task Force for COVID-19. He is also on the Technical Analysis Committee of the Government of Karnataka to tackle the present pandemic COVID-19. He is actively involved in writing articles for general public, providing his expert opinion to media houses and consultation to several states. I know the people of Karnataka are very much known to Dr. Giridhar Babu because so many debates are there over the television. People might have an evidence he speaks also. With this few brief introduction, it is over to Dr. Giridhar Babu, epidemiologist, for today's deliberation. Yeah.
Thank you, Dr. Alimuddin, for the kind introduction and also for inviting me to be part of the conference. And thanks to Ravi sir for informing you. So let me share my screen. Can you all see my screen, please?
Yes,
Sure.
next.
So, good morning, everyone. Today, I'll be discussing about the epidemiological features of SARS-CoV-2, and then also how the country is coping up with managing this epidemic. So, as of today, these are the number of cases we have, and most of the cases, as we know, nearly 84.7% of them are completely recovered. And there is a death, which earlier was dubbed as probably the highest fatality will be seen in the South Asia. This is how the initial projections were shown, but the South Asian region has proved as an enigma for most countries in terms of why the deaths are low. Let's discuss that for later. But if you see, the death per million is much lower in India. And
So sorry to interrupt, sir. Your slide is not visible here. The screen is there, but slides are not visible.
can you?
It's okay, sir. Now it's visible.
Oh, thank you. I think there's a different screen earlier. So, as I was explaining, the deaths compared to many other regions in the world are fewer in the South Asia, but in India in particular, this has been much of debate over the last few months. But deaths per million is 76.9. That means out of 10 lakh people in the country, we were seeing only around 76 deaths. If you compare that to the other countries, it is as high as 600, 500, depending on which country you are. So the deaths are probably 5 times higher or 6 times higher in the other country. The recovered proportion, if you say recovery rate, it is 84.7%. What started off around 3,000 tests per day, now we have reached nearly 1 million or more per day, and more than 80 million tests have been done in the country. So if you look at the population level, it is like 65,000 and above per million tests are done each day. 800 to 900 deaths per day is what the average deaths we are seeing now. But this is much less compared to even few weeks ago, which was above 1,000. If you compare with the other countries in the world, this is how we fare. Everybody keeps referring to the highest number of cases that the country is having. So let's get these statistics in perspective. If you see the cumulative cases, yes, we are ahead. And then if you just look at the number of deaths cumulative and also the deaths reported in the last 24 hours, each death has a name and there is a family behind it, and each death is very unfortunate. But as a population unit, these are fewer compared to the other countries. So globally, every day around 5,000 people die. Among that, nearly 900 and around that are the deaths in India, higher than United States, Brazil, and Mexico at current point of time. And number of cases per day, earlier it used to be around 90,000. It has reduced to around 75,000 and fewer, 65 per day. But we'll have to understand that detecting the number of cases wherever they are present, especially any circulation that is going on, is very important, because that is the only way we can reduce the deaths. Because only when you identify people, we'll be able to know who among them require hospitalization, treatment, and whatever supportive measures we need to take, especially oxygen has been life-saving. So this is how we can save. If you don't find, then you can't save. That's the most important. So we should not be bogged down by the number of cases that we report, but how many lives do we save? That's the most important part. So if you were to compare to the states, other countries in the world, you see where India is. Already some amount of celebrations are seen where people are saying India's worst is over and the peak has already been reached and its cases are coming down. If you see the India's scenario, the number of cases have definitely peaked. But despite of having more than 1 million tests per day, the number of cases or the growth rate at which the cases are detected is coming down. Is this the sign that the worst is over? We don't know. We'll have to find the new areas of, or all the dense areas are already infected, or newer cities, newer dense population pockets. All these things are very important in the days to come. And with the winter, whether we will see another surge, we really don't know. Having said that, we have not come down completely towards the bottom, so therefore we cannot say the worst is over yet. In terms of daily deaths, we never had a peak like we had seen in other countries, especially in the European Union or for that matter, United States. But there is also a problem of under-reporting, which people have been debating. My own sense is that under-reporting is an issue, but then it will not be as big issue that our death rates are similar to what's happened in the West. So we might have missed by some margin, but not really a big margin. So the tests are definitely increasing. And even if you look at the test positivity rate, it is falling down. So these are all as of yesterday. So again, when you look at the states, there are some states which report to be having higher number of cases, some lesser. And this I would want to explain by showing you this slide. Now, the first one, what you see in terms of blue shaded state maps, the darker the states are, it means the higher the number of tests done. So now, if you stay with me on only the dark shade in each map, the darker the color blue is, the darker the green color, which means whichever states are doing better tests are the states which are detecting probably higher burden of cases. For example, just stay with me in the Andhra Pradesh case, they are doing more than 1 lakh tests per million population. Therefore, they're also detecting more than 12,000 cases per million, and their deaths per million is 107. So what does it say? This is a fallacy that we see in almost every public health program. The states that have functioning surveillance system, the states that have better public health surveillance, the states that are focused towards improving testing rates And following syndromic approach, which Ravi sir clearly explained earlier, only those who have symptoms, if you test them, then the yield is really high. Those states will obviously have higher number of cases. It's a wrong thing to say that there is high burden of the state, COVID-19 only in few states, especially in the southern states. Actually, the fact is that these are the states which are reporting well. So reporting should not be confused for higher burden. And if you see the central belt where the testing rates are relatively less compared to the dark shaded states, the cases per million are also fewer, and also the deaths per million also fewer. So what is it? If you want to save lives, then you improve your approach of finding cases through syndromic approach, improve the testing rates, although it will increase the numbers of deaths, but then you're actually saving many more people because 84% is the recovery rate. It means that for every death that we save, we are saving more than 100 people. So that's the kind of approach one needs to follow. The case to infection ratio was nearly 100 in the first seroprevalence survey, which means for every case detected, there are 100 infections that we have not found. In the second round of survey, it has been shown around 26. For every case detected, there are 26. That means we have improved the testing tremendously throughout the country. These are the seroprevalence estimates in different parts of the country. Andhra Pradesh state shows around 40%, and the other states, including Delhi, are around 29%. If you were to look at descriptive epidemiology of the COVID-19 per se, you will see that most of the cases do occur in the productive age groups, which is between 21 to 40. And most of them are urban, rural is around 12%. Most of them are male. And if you look at the comorbidities, although there are problems in documentation, hypertension, diabetes, and liver disease are among the top 3. Even otherwise, if we look at the data, 1 in 3 adults will have hypertension and 1 in 10 will have type 2 diabetes mellitus. Therefore, there's no surprise that such comorbidities are also seen in the COVID-19 cases. So when you examine these in terms of states, I already showed this in the form of a map, but just to figure out which state is performing how, you will see that the testing rates are recently increasing in Uttar Pradesh, followed by Bihar, Tamil Nadu. These are the daily tests. So these are not indicative of real tests per million, because most of the states which are improving or increasing the test, I'll not say improve, increasing the test, are doing so by employing rapid antigen test. That is okay, as long as symptomatic negative or tested with RT-PCR. If that's not followed well, then the numbers are increased, but you're actually not finding cases. So that's not an effective strategy. And if you look at Tamil Nadu, they're doing only RT-PCR. So you have to have a combination of rapid antigen and RT-PCR to detect as many cases as possible and as many areas of circulation possible. In terms of daily deaths, Maharashtra still leads with a high number of cases, followed by Uttar Pradesh, West Bengal, Punjab. Daily cases, if you see, again, Maharashtra is showing a really high number of cases, followed by Karnataka, Tamil Nadu, Kerala, and Andhra Pradesh. So this is the next in terms of state-wise positive cases. The entire country we have shown in the red. Then you'll see that what is the total number of cases detected in each state. And as I explained earlier, this is dependent on the tests that are done in each state. For me, this is an absolute indicator of how well the state is doing its surveillance program. So if you ignore, although we should not ignore any region, but if you just want to compare the mega states which have higher population, you see that Delhi and Andhra Pradesh have the maximum cases per million. It does not mean that Delhi and Andhra Pradesh have more vulnerable people and the virus infects more people there. As Ravi sir clearly said, there's no problem in the seed, but the soil. So it's the people's behavior, which is same almost in the entire country. You cannot say people in Delhi and Andhra Pradesh are more vulnerable, but the higher number of cases means that the states made active effort to find more people with infection. That's all. So, and if you start going towards your right, and then you'll see that there are many states which are doing even the Indian average of 4,960 per million is not a great thing compared to just Andhra Pradesh, which is doing nearly 3 times higher. So this is an important indicator that we find areas of circulation so that we prevent deaths. And this is similar, this is proportionate to the tests per million, and deaths will also be there. So once you find cases, I'm part of this hospital inspection committee also. What we see, unfortunately, is that if a person with severe illness comes to the hospital, there is a tendency to keep referring them to other hospitals. But ideally, if you find more cases, you'll also find people who are vulnerable from this disease. So therefore, such people would require treatments. Just because you're identifying them, you'll also report higher deaths per million. So there is a taboo. There's a stigma of the disease. There's a stigma of labeling deaths due to COVID. And as a result, there is underreporting, especially probably higher in the states which are not testing well, which are not detecting the areas of transmission well. This is the case fatality. Case fatality indicates how good our treatment is, and it is improved after the new treatment provisions are made. Shashi sir will explain what are the modalities, but the states that have improved their clinical performance have reduced the case fatality rate. So the lower the case fatality is better, but to know the total mortality, it is better to look at death per million rather than the case fatality rate. And this is no brainer. Every state will have a good amount of recovery rate over a period of time. So this is just for completion's sake that I have put here. So what are our actions, and what is that from the epidemiology that we need to understand? The pandemics will come and go, but what we need to understand is, do we have a resilient system to detect any outbreak that happens? And India has a very ambitious program called Integrated Disease Surveillance Project, IDSP. So it's time to strengthen that at every district level. And the action should be taken at the district level itself in terms of looking at the data and then taking action. The lab testing and strengthening should occur throughout the country. And the risk communication is very important. Now you see that most of the media houses are actually spreading rumors and wrong information, including our own social medias, which have become the main sources of misinformation. So as the Director General of WHO said, we are fighting two pandemics. One is COVID-19. The other one is the pandemic of misinformation. So we need to battle both. And for that, risk communication has to improve. Hospital preparedness is most important. We have seen only public health system is the strongest one. So at the peripheral level, we need to strengthen primary health care and integrative approach. Not just the one system of medicine, but all systems of medicine should play their role in prevention. But when it comes to critical care management, our capacity needs to improve. The logistics have to improve, and the capacity of the staff needs to improve. So I wouldn't go into the other details, which probably Shashi sir will be able to clearly delineate. But what we saw is these pandemics induce stronger problems, most adversities in the people who already have the comorbidities. So therefore, if you continue to neglect the chronic diseases, and if you do not build a system I'll just give you an example and then I'll go to the next slide. as I said earlier, 1 in 3 adults have hypertension. We do not have a screening program to detect all the people with hypertension. But we have insurance programs for reimbursement if somebody gets a heart attack or a stroke. What does that mean? It means that we are okay to invest in an insurance program when somebody gets into serious complication, whereas we are not investing even 1 rupee. So 2 lakh is the limit that you give for myocardial infarction, heart attack. But we are not spending 2 rupees on antihypertensive medications. This has to change. These pandemics will inflict only pain on those who are already affected. So these diseases have to be catered. That's the learning that we have. So again, I'll not go into the details of severity of illness. What we are really worried about the severe illness, which we need to take care. Moderate have to be monitored so that they don't go to the severe. And Ravi sir has explained on vaccine trials. I'll not go into the details of this, but then there's no hope that we'll get a vaccine in next few months. It will take some time. The only vaccine we have is mask as of now. Again, there's a lot of misinformation about the mask. Avoid 3 C's, which is crowds, closed spaces, and close contact settings. That's the vaccine for now, apart from using the mask for some time. And although these are the initial signs of the curve going down, let's be careful with these measures so that we don't see the similar second peaks some other countries are seeing. As a country, we have done so far well, despite of all the different narratives that are being told. People gave doomsday predictions for the country, but we are holding up and we have shown. But 900 deaths per day is not something very welcoming. So we should be able to prevent as many as possible. So these are the measures. And now people want to open schools, colleges, and workplace measures will have to be strengthened. Therefore, this is the time for us to be even more careful. Earlier, we were hiding away from the virus. But now that we are freely at least meeting, then we should be able to control the kind of control settings that we need to follow have to be strengthened. So that concludes my presentation, and I'll be happy to answer any questions that you have.
Thank you, sir. It is a question to Girdhar, sir. Generally, there is the common public will ask the question to the consultants of the doctor. When this pandemic is going to be complete, finished, why would have? I mean, they are expecting something that the flattened curve. With reference to the epidemiological term, that R value, how you are going to say that what is the status of the India in comparison to the other countries, particularly even in India also, which are the states which are leading towards the -1 R value. Can you enlighten upon this one, please?
That's a good question. So R0 for the country has been on the downhill for the last 2 months now. So I can give you the state-wise, but at least Andhra Pradesh has already gone less than 1. But the other states which are seeing the surge in cases now in the southern India, they also have reached around 1, but have not gone less than 1. Currently, for the entire country, it is around 0.98, but that's just an average of all the states. And it has progressively gone down. So although the R0 is going down, we'll have to be careful of the areas which are in terms of getting the new infection now and newer areas now. So this is early, but at least the evidence shows that currently there are states which are above 1, which is West Bengal, Tamil Nadu, Chhattisgarh, Assam, Rajasthan. Karnataka is 1.11. Nagaland, Manipur, Ladakh, Meghalaya, and Kerala. Kerala has 1.27. So this is regarding R0. Flattening is a term that has been introduced and has been much anticipated. I wouldn't say flattening in the sense that suddenly there will be a drop. There might be a plateau for some time, after which it will drop.
Thank you, sir. Thank you very much for the wonderful deliberation with reference to the epidemiological point of view. Thank you for joining with us. Let me take a privilege to the next speaker who is standing in line. We have a very wonderful person, Dr. B.L. Shashibhushan. He is graduated MBBS from Kempegowda Institute of Medical Sciences, Bangalore in 1993, and MD Pulmonary Medicine from Jawaharlal Nehru Medical College, Belgaum during 2003-2006. Sir will be having a more than around 20 years of experience, teaching experience. Presently, Dr. B.L. Shashibhushan is working as a professor and head of the Department of Pulmonology in Bangalore Medical College and Research Institute, Bangalore. This is one of the prestigious institutions in Karnataka and one of the reputed institution in India in medical education. Dr. Shashibhushan, sir, is like the other members of our today's webinar. He is also the member as a technical expert, Karnataka State COVID-19 Task Force Committee. He is a nodal officer for Center of Excellence for the Department of Health and Family Welfare, Government of Karnataka. Doctor is also a member as a technical expert for treatment protocol, Rajiv Gandhi University of Health Sciences, Bangalore, for particularly the COVID-19 Technical Committee. Dr. Shashibhushan is the nodal officer at the Victoria Hospital. This is the dedicated COVID care center as in today in Bangalore city, Bangalore. Technical head for the Victoria Hospital, dedicated for the COVID-19 hospital, Bangalore, the 550 bedded hospital, and he's in charge for the COVID-19 screening OPD. I'm very glad to call Dr. Shashibhushan is he's moderated several programs, conferences, webinar. I'm very happy to inform my august audience and the panelists. Dr. Shashibhushan organized as one of the conference, SleepCon 2019, the recent past only. This is first of the Karnataka state level conference of sleep-related breathing disorder. This is a wonderful conference. So many people are attended there, and this is need of the time also. I'm very happy to have this. Dr. Shashibhushan is the organizing secretary for the one successfully conducted this program. With this few words, it is over to Dr. Shashibhushan for your presentation.
Thank you, sir. At the outset, I would like to thank Dr. Mohammad Alimuddin for having invited me on giving a presentation, and also Dr. Ravi sir for having told you about me. As both Dr. Ravi and Dr. Giridhar Babu has spoken about the COVID, certain things have been little overlapped. What I'm going to talk to you will be on the clinical manifestation and management. What is at the state level and the country level what we are doing. So I have this disclaimer regarding the recommendation of the guidelines. Few points I would like to stress upon is when a patient who comes, we need to define whether they fall into influenza-like illness, what is called as ILI, or a SARI, severe acute respiratory illness. ILI is defined as a patient who comes with a respiratory infection with fever of more than 38 degrees and cough, and the onset should be within the last 10 days. So these are the suspected. What we are going to say is about the suspects about the COVID patients. SARI is same, who with the following things requires hospitalization is called as SARI. And these are the two categories of patients who needs to be tested. So this again regarding the respiratory failure, and just to brief upon this, it represents the failure of the lung to maintain an adequate gas exchange. Basically, ABG abnormality is what we are seeing in a respiratory failure. So a patient with COVID, what are the common symptoms? Observe how common symptoms he comes with. He can have fever. Fever is a predominant symptom. 87.9% is a predominant symptom. As both Dr. Alimuddin and Dr. Ravi sir has told that any system is involved in the COVID. So they may come with the fever, dry cough, fatigue, sputum, shortness of breath, GI symptoms, neurological symptoms. I'm stressing here more related to the respiratory part. What I told is they may come with smell and taste disorders, conjunctival congestion, hemoptysis, any presentation, any symptoms coming to a doctor should be suspected as a COVID at the present time. So once a patient comes, how we categorize these patients? We categorize basically for the management part. So basically it is being divided into group A, group B, and group C. What is group A? Group A is patients who are asymptomatic patients or patients with a mild symptoms, and their respiratory rate is less than 24 cycles per minute, and SpO2 is more than 94% in the room air. And group B are symptomatic who have mild to moderate pneumonia symptoms. They may not be in respiratory distress. So the criteria for that is respiratory rate is around 24 to 30 per minute, and his oxygen saturation at room air should be is around 90 to 94. These are the patients whom we need to target. Target in the sense like early intervention, early treatment, so that we can reduce the mortality. Group C is a severe form, severe pneumonia, whose respiratory rate is more than 30 and SpO2 is less than 90. And even on oxygen, the saturation is around 94%, and he may be in a septic or a ARDS shock. So these are the patients. Group C is one patient who most likely will go into the ICU, and the outcome of these patients are really not good. Like, so what are the categories, clinical categories we need to know about these patients? As I told you, see, it's the same thing, like mild, moderate, and severe. As per the guidelines, critical and these are the critical patients are those patients whose worsening symptoms, they come with a very bad chest X-ray. They could be opacities, infiltrations, who most likely go on a ventilator. So they may also land up in septic shock. So once a patient who comes, what are the investigations you would suggest for these patients? So in mild category, we ask for a CBC for them, because if it is a diabetic, and new diabetes can also happen in COVID situations. So D-dimer, if you are starting, because most of the time, as Dr. Ravi sir was telling, they may have thrombotic phenomena. So basically, ideally, initially only we need to catch them and find that their D-dimer values are increased. So that is the reason in our guidelines we are suggested for even a D-dimer values in a early mild cases. If you are starting a drug called favipiravir, we ask for a renal function test and a serum electrolyte and serum uric acid. Along with these tests, moderate to severe, what is basically the other tests called LFT, RFT, RBS, serum electrolytes, 12-lead ECG, chest X-rays, and what are the alarm markers? Basically, alarm markers are, these are prognostic values like ferritin, LDH, procalcitonin, troponin I, because the myocardial, they could be myocarditis, myocardial infarctions. To check for those readings, we ask for troponin INT values, PT, INR values, ABG is routine. If just CT thorax is available, try to get a CT thorax wherever possible, because in our institute we are getting CT thorax for most all the patients, most of the patients, especially in the moderate patients. And blood cultures. Initial days we didn't were not getting, but over a period of time we started getting blood cultures to find that if there is a secondary infection. That was the reason, and so that accordingly antibiotics can be started. IL-6 values, serum cortisol levels, echocardiography. Along with these things, like Ravi sir in one of the slides he was mentioning, there could be an overlap of IgM and RT-PCR positive. So we can know if we can get an antibody testing IgG, IgM at this level also. So what are the other in severe critical apart from these things? So apart from the routine, what in a critical patient is, we ask for a serum magnesium, serum calcium, pro-BNP again to look for the cardiac issues, CRP values. CRP can also be got in the moderate cases also. So once you get all these tests done, when do you repeat in according to the situation of mild, moderate, and severe? In mild, usually we don't tend to repeat, because these are the people who are going to be at home or in home isolation or a CC, COVID care centers. So that's the reason there is a no repeat of these tests. Moderate to severe are the patients who are in the hospital. So basically, we try to repeat these tests every 72 hours. These whatever test in a routine Daily testing is complete blood count, LFT, RFT, and ABG in both moderate and severe. Every 72 hours when it comes to markers, so which helps us in the prognostic values. At the time of discharge, certain tests, CP or serum, even if there is a fall in reduction in the values of a D dimer or serum ferritin levels, you can suggest for a discharge, because symptoms is not the only criteria. I'm going to touch upon the discharge criteria also. So on certain, and RT-PCR is only done, a repeat RT-PCR is only done in patients. Who are the patients who are high risk? So comorbidities. So age is one comorbidity. Age more than 50 is a comorbidity. Hypoxemia, who is less than 94%, you need to look for them. See for the lymphopenia. Basically, neutrophil lymphocyte ratio is very important to predict the prognosis of the disease. A simple test like a complete blood count will give you a lot of information. The patient is ischemic disease. You have to see if he is having tachycardia. And other comorbidities like diabetes, hypertension, any chronic lung disease like COPD, asthma, tuberculosis, chronic disease, chronic liver disease. These are the patients who are what we are seeing in our most of the time. It is a chronic kidney disease. Chronic kidney disease. In a routine dialysis, the chronic kidney disease patients who undergo dialysis, the mortality among these patients itself is 30%. In a situation with coexisting COVID, definitely the mortality will increase. So this is where we need to, get these people aware, especially nowadays. Comes to malignancy also, it is the same. Obesity is another very important comorbidity we are missing, because a lot of patients die because of obesity in COVID infection. So general measures, you do with a patient who's been diagnosed to have COVID positive. Basically, you give them supportive care in relation to the nutrition of their food, then ask for a finger pulse oximetry, monitoring of his heart rate and oxygen saturation, if it is at a home or a hospital, both ways. So even in a home isolation, these are the things you need to have with you, with a pulse oximeter and BP apparatus. Start oxygen with mask at saturation of 90%. These are certainly very important. Sometimes some of them do not want to get a pre-existing lung disease. A patient who's already on an oxygen, home oxygen on a concentrator. So these are the patients we need to monitor, like interstitial lung disease or a COPD, or who are dependent on oxygen. So we need to ask them to continue these the oxygen concentrator and routine medications, what they're what they're using. So another most important is high flow nasal cannula to be used if there is a failed oxygen therapy, and non-invasive. This is a part of the treatment in the ICU. What we are trying to do when the patient don't respond with a routine oxygen therapy, we try to give them non-rebreathing mask, which can give about up to 15 liters of oxygen. If he's still not responding to that, then you use this high flow nasal cannula, where you can give oxygen up to 60 liters, and a non-invasive alternative with a non-invasive. Some of the patients require a non-invasive ventilation like a BiPAP or a CPAP, who basically respond very well with an underlying obstructive sleep apnea. So all these measures are very important. And once a by a counselor or psychologist or a psychiatrist is very important, both to the patient. We need to ask the psychiatrist to counsel the patient as well as his attender. Daily talking to them will improve their morality every day. That's very important. And the routine normal feeding, and there is no dietary restriction for a COVID patient. You can have anything, or if he's without a comorbidities. And hydration, again, IV fluid is very much indicated in a case of especially elderly people who are more than 60 years. We need to give them good hydration. So if there is no hydration, definitely there will be this electrolyte imbalance. Electrolyte imbalance is one of the common things we are seeing, and hyponatremia, common. What we are seeing is this has to be attended so that the caring or a mortality. These are the things which needs to be intervened. So mortality can be reduced, and our target of blood sugar should be around less than 180 milligram per deciliter. If the patient is already on ACE inhibitors and or ARBs, they need to be continued. There is no specific, I mean, you don't have to stop these medications, and try to avoid non-steroidal anti-inflammatories and use paracetamol unless it is absolutely necessary. Basically, we try to avoid nebulization. In case there are some patients who are on home nebulization, get admitted. Preferably, if there is a negative pressure ventilation, we try to continue using nebulization. If there is no setup like that, probably we prefer them by giving them meter dose inhaler with a spacer. Total culture, blood culture is done to check for if there is a secondary bacterial infection, and specific antibiotic is being given. Coming to the treatment part, in mild case, so what are the drugs we give in a mild patient? Patients basically can be at home isolation or a COVID care center. So these are the drugs suggested as per our state guidelines. We can give tablet hydroxychloroquine 400 milligram BD for day 1, followed by 200 milligram BD for day 4. And before we start hydroxychloroquine, the basic investigation, as I already told, ECG is a must for these patients. So if there is a QT interval prolongation, if it is more than 480, it is contraindicated. In such a situation, if the patient is contraindicated and mild symptomatic, then we start the patient on favipiravir. This is antiviral drug. Most of these, whatever we are giving in mild, are all has shown some antiviral activity. So favipiravir on the first day, 1800 milligram BD for day 1, and followed by 800 milligram BD for next 6 days, total of 7 days. So these patient, in case they are symptomatic, they will go to dedicated COVID healthcare center. They may not be in CCC or they may not be home isolation. So in case a situation arises when they're both HCQ and favipiravir contraindicated, the next alternative option what we have is doxycycline 100 milligram BD for 5 days, along with tablet ivermectin 12 milligram OD for 3 days. And we have suggested oseltamivir, keeping in view of H1N1 pandemic, situation because of condition. So because we are not testing for the H1N1, the present condition of the COVID. Initial days we used to do, we have seen some of the patient H1N1 positive with COVID. So that was one of the idea of using oseltamivir in our guidelines in mild, moderate, severe cases. And if your D dimer is on the higher side, so in a mild case, we may have to start them on enoxaparin. This is a low molecular weight heparin, which is given 40 milligrams subcutaneously for 7 days. So apart from these supportive therapy, what we are giving is tablet zinc 50 milligram OD for 7 days, then tablet vitamin C TID for 7 days. N-acetylcysteine is a mucolytic. Basically, it is a mucolytic and antioxidant effective. Why we have suggested here? Because because of the COVID infection, there could be thick tenacious secretion. So this basically will lyse the secretion. So initially we are targeting at the initial levels only. So N-acetylcysteine is given for 3 times a day for about 5 to 10 days, depending upon the situation. So on the right hand side, I have mentioned certain contraindications for the following drugs. Coming to the moderate cases, what are the treatment in moderate cases? When it comes to moderate cases, we have specific antiviral called as injection remdesivir, which is given for totally 5 days, 200 milligram on day 1, followed by 100 milligram for the next 4 days. So in case if remdesivir, now the availability of the drug injection remdesivir is available everywhere in the country, and in a taluk hospital or a small PHCs, you can name it. The government has procured this drug and have been distributed everywhere. In the initial days, we had problem in getting this drug. So in case they don't get this remdesivir, we have suggested hydroxychloroquine to continue. And the next most important thing, the other drug is methylprednisolone or dexamethasone. They again, these two injection remdesivir and methylprednisolone, the indication for these two is the saturation should be around 90 to 94. This is the early initiation of remdesivir and methylprednisolone. When the saturation is 90 to 94, the patient tend to do very well when you intervene at the early time. So this is very important. The timing of these two drugs. Again, CRP is one important indicator where whether we need to use the drug methylprednisolone or dexamethasone is important. And to rule out a secondary bacterial infection, we ask for a procalcitonin levels. So usually in bacterial infection, procalcitonin levels will be high. So once you are sure that there is no bacterial infection, you can start methylprednisolone, and the response is very good, what we are seeing in our setup. And other drugs, as I already told, is the low molecular weight heparin to continue. And in case the patient don't respond to even remdesivir or a steroid, the next most important antiviral is the convalescent plasma therapy. So this is again, usage of convalescent plasma should be within the first 10 days of the infection. We are trying to use in moderate cases. We have studied, we have done a trial for both moderate cases and severe cases. The response in severe also is not really bad, but the patients who have recovered in severe cases also. But our experience on the usage of convalescent plasma therapy is really good. The timing, again, we have, as per the ICMR report, they have found out that it's not really happening up to their expectation. But in our setup, or most of the places, what we have heard, the timing of usage of convalescent plasma is very important, and it has responded, and patient have come out of the orange zone, what we place the patients, and they have not gone to the basic ICU. We have prevented them going into the ICU. Even in the ICU, prevented the patient going on a ventilator. So these are very important things I'm stressing on when it comes to moderate cases, and certain contraindications for the usage of remdesivir. You need to get a LFT, RFT before we start them. Remdesivir we have used in a 83 years old whose GFR was 30 ml per minute, but he has responded well and gone home. We have discharged him. So he's really doing well. Age is no criteria for the usage. We just have to monitor the drug, get a routine RFT, LFT to be done when you give these drugs. When it comes to severe critical cases, sometimes we may have patient, if the patient has not received remdesivir and directly come in a critical situation, so we tend to give remdesivir. Along with remdesivir, the most important another drug is injection tocilizumab. This is again a double-edged. Injection tocilizumab and itolizumab are two anti-IL-6 drugs. This is a double-edged sword-like situation we are dealing with when we start using tocilizumab, because it may worsen a patient condition or may be detrimental, but in certain situations these can be very effective. So what are the indications when you want to start tocilizumab? See, IL-6 values, the interleukin-6 value should be more than 100-fold. So that is an indication. There are people or doctors using IL-6 values about say 8, 10 and all. So here it may be detrimental. So there should be judicious use of tocilizumab indications. And apart from the steroid usage, the low molecular usage, what most important thing what we have found out is the prone ventilation. Prone ventilation can be with oxygen or on a ventilator. Either of the prone ventilation can be done. It basically helps the patient recovery. In sorry, I put these slides again mentioning the same thing like. So all these investigations are approved recently by the DCGI and should be used with caution. And before we start tocilizumab or remdesivir, we need to take an undertaking from the patient, and with their consent. Patient consent or a patient attendant consent is very much important before we start these drugs, because all these are investigational therapies. There is no specific treatment for this. Coming to the discharge policies, we have a discharge policy. When do we want to discharge a patient who's at home. Asymptomatic patient can be at home or in a hospital. What is the criteria for those things? Asymptomatic shall be discharged 10 days after the positive test report. If he's asymptomatic during his stay. Earlier, this was asymptomatic were admitted in hospital also in earlier days. But now we have specifically seen that these patients don't get hospitalized. Either they are in the CCC or a home isolation. So there is no repeat test required for these patients to get it done. Mild to moderate cases, they shall be discharged 10 days after symptoms. That is, there is again no need for a repeat RT-PCR test for these patients. And the last previous 3 days, there should be asymptomatic. 3 consecutive days, they should be asymptomatic. If it for a hospital admission, 10 days of admission prior to the 3 days of asymptomatic, they should be asymptomatic. You can discharge them. If they have symptoms prior to their admission, then even at the 7th day, say 3 days before they have symptoms, they come to you to the hospital on the 3rd day, you can discharge them on the 7th day. This also is one indication. And your repeat inflammatory markers, like there should be a reducing trend, decreasing trend in these. That's another indication to tell us whether the patient is responding or not. So there is no need for a RT-PCR done. Only in a patient who is in the ICU, who is critically ill, recovered, this patient is the only patient we require a repeat RT-PCR. It can be either of the RT-PCR, a CBNAT, or a TrueNAT test can be done to see. In case it turns out to be positive, then it has to be repeated 72 hours. We have a situation where we have done a RT-PCR for an elderly man who 91 years who continued to be positive every 3rd day for almost more than 11 times. So that our experience is there. Probably Ravi sir would agree that it is probably the remnant, the remnants of the RNA for and that was the reason the repeat RT-PCR test was not done in a mild to moderate patients. So if they turn to be positive, it may be a remnants. The only important thing what I am stressing here would be on the symptoms. So patient who's symptomatic only should be given more importance rather than the blood reports. When it comes to home isolation patient, they should be home isolated for 17 days. So here at least 3 days they don't require they should not have any symptoms like fever, maintain saturation without any symptoms. They don't require a RT-PCR. On the 18th day they can start going to work. So these are some of the things I have briefed upon regarding the treatment aspects. I thank Dr. Alimuddin again for giving the opportunity. Thank you.
Thank you, Dr. Shashibhushanji. Thank you. Here, one of the questions generally the people are expecting is, in the first week of the infection, how far it is justifiable to use steroids among the even mild to moderate cases also. The general practitioner, they started using steroids in the first week of the positivity of the COVID cases, irrespective of the nature of infection, whether it is a mild, moderate, or severe. Because as I was looking forward on the news, initial phase administration of steroids is not suggestible. Please enlighten upon this point, sir.
The role of steroid in mild cases is detrimental. So it is not indicated. Detrimental in the sense like it can complicate even to such an extent that death can happen. So the guidelines as per the recovery trial, whatever the trial, on that basis, it is proved that it is specifically for moderate to severe patients only, not for a mild patients. So that is the reason I've stressed two very important things. One is the symptoms, one is the saturation levels, 90 to 94. If they fall in the range of 90 to 94, and the CRP value, C-reactive protein values, which helps us in starting the steroids.
Okay, thank you, Dr. Shashibhushan, for joining with us. Thank you. Let me invite the speaker who is on the line. We have a very the next speaker is Dr. Manjunath M. Venkataswamy. Presently, sir is working as an assistant professor, Department of Neurovirology, again from National Institute of Mental Health and Neurosciences, Bangalore. Since 2017, Manjunath sir is working, and he did his MBBS and PhD from neuromicrobiology from NIMHANS only. Postdoctoral fellowship 2005-10, Department of Microbiology and Immunology from Albert Einstein College of Medicine, New York. He was associated in the research since 2010 to 12 in the same Albert Einstein College of Medicine, New York. When he came back to India, a brief stint at Ananda Diagnostic Laboratories, Bangalore, as a senior consultant, he extended his services in molecular and flow cytometry diagnostics. Since 2012 to 2017, as a senior scientific officer, worked in the neurovirology unit of National Institute of Mental Health Neurosciences, Bangalore. He's a coordinator for flow cytometry core facility at NIMHANS. This is one of the prestigious Institute for Neuro Health and Mental Sciences in India. Actively involved in COVID-19 diagnostic laboratories. The sir was awarded in 2010 as a Young Investigator recognition, Bill and Melinda Gates Foundation for significant contribution towards HIV vaccine research. Sir has one US patent on incorporation of NKT cell activating glycolipid vaccine adjuvants into bacterial cell wall, the postdoctoral work in fact. His areas of research interests are immunology of infectious disease, vaccine adjuvants, diagnostic virology, immune dysregulation in neurological and psychiatric disorder. Sir Manjunath sir is going to talk about the current status of vaccine. Over to Dr. Manjunath.
Thank you, sir. Thank you very much for your kind introduction. And I'm really thankful to the National Institute of Unani Medicine, the director and Dr. Alimuddin for this invitation to present here. It is indeed a privilege to be presenting among all the stalwarts in the field. So is my slide seen, sir?
No, it is not. Please. It's not visible. Of course, your screen is visible, but not the slides. Maybe you have a different screens are there. Go to the presentation.
Yes, sir. I am in the presentation.
No, here we are unable to see this PowerPoint presentation, sir. It's a whiteboard, in fact.
Right. Okay. Right. Now, sir?
Yeah, yes, sir. It's okay.
Thank you, sir. Thank you. Sorry for it.
Thank you.
So my topic for today would be COVID-19 vaccine current status. So the previous experts have already mentioned to you, what the status of the vaccine is briefly and what is to be expected in the next few months. Yeah. So what is the goal of vaccination in general? So we have had many successful vaccines. In fact, human beings have fought and eliminated several infections. In fact, deadly infections like smallpox, with very successful vaccines being developed. And everywhere the goal of vaccination is to create immunity in the host to prevent disease if the infectious pathogen is encountered. And the main advantages of this is to mitigate the public health, economic, and societal impacts of the virus. A vaccine is urgently needed in the context of COVID-19. Usually, vaccines are designed to induce antibody responses that target viral proteins involved in cellular entry to neutralizing conditions. And SARS-CoV-2 vaccine development was initiated in early January 2020 when the genome sequence became available. In fact, it was just very few weeks that after the pandemic became evident across the world. So before even it started spreading across the world, the genome sequence was available and vaccine development was initiated. Please don't mistake me. I just wanted to introduce just the basic components of the immune system because vaccination is about inducing immune responses. Like Professor Ravi mentioned, the innate immune response is the most important for preventing the initial infection stages of the virus, so that we get rid of the virus very early on. So here, this slide shows you the very basic components of the immune system. On the left is the innate immune component, and the right is adaptive immune component. So all these different cells, the dendritic cells, mast cells, macrophages, natural killer cells, and the various other granulocytes form the innate immune component, cellular component. And on the right, the adaptive immune cells are the T cells and the B cells. So with vaccination, we are primarily targeting educating of B cells and T cells so that they remember the exposure to an antigen, and if a similar antigen from the pathogen is encountered by the body in the future, these B cells and T cells are already equipped to get rid of the infection without us even getting noticing the infection. And in the interface here, you have these other cells, the gamma delta T cells and the natural killer T cells. A few other cells have been added to this list now. These are called innate T lymphocytes. There are some mucosa associated invariant T lymphocytes and innate like lymphocytes, which are newly identified T lymphocytes, which play a very important role in bridging the innate immunity to the adaptive immunity, so that whenever an innate immune system recognizes exposure to a new pathogen, the signals are efficiently transmitted by these cells to the adaptive immunity. In fact, these cells can be double-edged swords. They can either be protective in nature or also may result in more immunopathogenesis. So with that brief introduction, I'll just move on. So, so you're all very familiar with this. So whenever SARS-CoV-2 virus infects an individual, so you know, like the virus has many different proteins. Dr. Ravi has already mentioned to you. So one primarily important protein for the virus is the surface protein, or which is called the spike protein or the S protein. It helps the virus to bind to the ACE2 receptor on the host cell. The S protein binds to the host cell and then enters the epithelial cells. There you have the viral genome being released into the cytosol, and then you have the viral RNA being translated into proteins, and virus is assembled, and then the virus is released by the epithelial cells. And now this virus is ingested by the innate immune cells or the antigen presenting cells, the APC, for example, a dendritic cell. So the dendritic cells are very efficient in inducing immune responses, either to educate T cells or B cells. So when these dendritic cells present antigens from the virus to the T helper cell or the CD4 T cell, that in turn helps in activating of B cells and activation of cytotoxic CD8 T cells. The cytotoxic CD8 T cells are equipped to kill infected cells. So they destroy infected cells by apoptotic death, and antibodies in turn, which are produced by B cells, will cause neutralization of the virus, thereby preventing. So the antibodies bind to the surface protein, for example, here, and prevent the attachment of the virus to the host cell receptors. And the most important component of this immune response is generation of a memory pool. The memory B cells and the memory T cells are generated, and they live in the body for several months to several years, depending on the nature of the antigen and the quality of the signal, the activation signal that they receive. And all our successful vaccines are dependent upon this particular component of the immune system, that is generation of efficient memory B cells and memory T cells. In fact, so you see that a natural infection can lead to generation of memory B cells and T cells here, right? So in vaccination, this is what we are trying to target by educating the immune system to generate memory B cells and T cells. And to shift gears, so if you look at the traditional vaccine development versus the accelerated vaccine development that we are witnessing with respect to COVID-19, if you take the example of a traditional vaccine development, you have first few years where there is, it's more exploratory in nature, where you're trying to identify what are the protective antigens, the crucial antigens of the virus against which we need to develop immune responses by vaccination. So a lot of time goes in that. And in the next 2 to 4 years, there are a lot of toxicological studies, preclinical studies in animal models to look at the efficacy of these vaccines and their safety levels. And then the application is submitted to get an interventional new drug approval. And then the vaccine moves into the phase 1, wherein less than 100 individuals are evaluated. The vaccine is administered to less than 100 individuals and to test for safety. It takes average about 2 years. And then we move on to phase 2 trials, wherein few hundreds of individuals are administered the vaccine to look at immunogenicity, the optimal dose and the optimal regimen, the route of administration, how much vaccine to be given. All those things are studied during the phase 2 trial. And if there are encouraging results here, the vaccine moves on to the phase 3 trial. Again, it takes about 2 to 3 years, where thousands of individuals are administered the vaccine candidate to look for efficacy and safety again. So once there is encouraging data from all these different phases, then applications are submitted to regulatory agencies, like it might be a FDA or a European medical agency or any local regulatory agencies for approval for large-scale production and distribution. So overall, the whole traditional development of vaccines may take up to 15 years on an average, although it could be a few years lesser in some cases. Against this picture, we are trying to squeeze in the COVID-19 vaccine development within a span of, we are trying to fit in all these different phases of vaccine development into 10 months, between 10 months and 1.5 years. So, wherein one important advantage was we had some very good knowledge about protective antigen, that is the S protein of the virus, through earlier research on the SARS-CoV-1. If you remember, the SARS epidemic occurred in between 2002 and 2004, and luckily the virus got eliminated. Although some vaccines were developed, they did not progress further because there was no need to develop them in large production phases. And also MERS-CoV again emphasized on the importance of the MERS coronavirus, basically, importance of the S protein for the virus. So with that knowledge, we were able to directly identify the S protein of SARS-CoV-2 as the candidate vaccine antigen. So we were able to kind of quicken the whole process. And also the phase 1 and phase 2 trials were overlapped in many cases. And quickly, at least a few vaccines have progressed to phase 3, and licensing applications have been already submitted to regulatory agencies. So in all possibility, we are expecting vaccine to be around somewhere between March and August of next year. So to give you an example, the different vaccines that have been developed by human beings over the last many decades, if you just take polio vaccine, one of the very successful vaccines in recent times, in the last 2 centuries. So you'd see like an average of 60 years was required to come up with the very good polio vaccine, which helped us eradicate polio in the year 1979, at least in the USA. And then Ebola again took about close to 15 years to develop a successful vaccine. And in SARS-CoV-1, there's, although the epidemic occurred in 2003, there has been no successful vaccine in the last 17 years. One primary reason being, there has been no commercial interest because the SARS-CoV-1 virus by itself got eliminated from human populations after 2004. However, with MERS vaccine, the efforts are still going on. Again, that vaccine development is suffering because of a lack of enough funds to push this development. And with respect to SARS-CoV-2, like I told you, there are a few vaccines, and in fact, one of the first vaccines entered the first clinical trial in as early as March. That is just after 3 months after the pandemic was identified. So, so we are expecting about 1.5 years maximum to have a vaccine to be administered in human beings. So as it was mentioned, more than 180 vaccines have already been developed, and they're all at various stages of development, either preclinical or phase 1, phase 2, and phase 3. So many vaccines against SARS-CoV-1 were developed in the preclinical stage, and 2 of them actually progressed to phase 1 clinical trials. This is SARS-CoV-1. Okay, further development stopped as the virus ceased to affect human population, and it did not re-emerge after 2004. So vaccines against MERS coronavirus are actively being developed and have been supported by this important organization, which helps vaccine developers with funding, which involves millions to billions of dollars. There's a coalition for epidemic preparedness innovations. In fact, one of the Indian institutes, which is in the forefront of vaccine development in India, has been made part of this particular coalition recently, which I'll mention to you later. Okay, so so although we did not have successful vaccines for SARS-CoV-1 or MERS-CoV-2 yet, the important advantage is preclinical studies with SARS-CoV-1 and MERS-CoV have clearly revealed the antigenic target for vaccine design, and that antigenic target is the spike protein. And the COVID-19 vaccine development has moved at record speed, with, as I said, one phase 1 clinical trial already starting in March 2020, and more than 180 vaccines in various stages of development. Phase 1-2 trial data is already available for several vaccine candidates, and many have moved into phase 3 trials. Data suggests that effective and safe vaccines might be available in months, if not years. Development of vaccines traditionally against human coronaviruses has been of low priority, as they cause mild disease, and also because SARS-CoV-1 got eliminated naturally, and MERS-CoV vaccine, MERS-CoV has not really spread other than the Middle Eastern areas. And SARS-CoV-2 is a beta coronavirus closely related to the SARS-CoV-1 virus, which occurred between 2002 to 2004. Okay, so when does vaccine development really progress? Why did SARS-CoV-1 vaccine development get stuck after initial clinical trials? That is because the data from all phases, it should look good. Otherwise, it's not encouraging for vaccine manufacturers to go ahead, and it should be associated with a low risk of failure. If vaccine manufacturers are going to invest huge amounts of money and effort, then the risk of failure should appear low. And whether there is an available market for the vaccine manufacturers, that is an important determinant for whether vaccine development progresses until it reaches the clinic. So with respect to SARS-CoV-2 vaccine, as it was already mentioned, the spike protein is the most critical antigenic target. It is a primary target for vaccine development. So it forms this protein is found in the surface of the coronavirus, as you can see here. And in fact, the receptor binding domain, which is shown here, which is part of the S protein, is another candidate antigen. Many vaccines are based on just this particular RBD, or the receptor binding domain of the S protein. Vaccines are in clinical trials based on RBD, along with vaccines on S protein. So what is S protein? It's a large surface protein which helps in virus attachment to receptors on host cells and helps in membrane fusion. The S protein of both SARS-CoV-1 and SARS-CoV-2 binds to the ACE2, which you are all aware already, and helps in the virus being endocytosed. That is, the virus is being taken into the cell. So now the fusion of the viral and the endosomal membranes results in the release of viral RNA into the host cell cytoplasm. So this kind of indicates antibodies against the S protein, particularly the RBD, or the receptor binding domain, can inhibit viral attachment and neutralize the virus. So based on the earlier available knowledge from SARS-CoV-1 and MERS-CoV, the S protein was identified as the antigenic target of SARS-CoV-2 in initial stages. So S protein is also a strong target for CD4 T cells, while fewer CD8 T cells are induced by natural infection with SARS-CoV-2 in general. So which means to say the S protein can induce antibodies as well as T cell immunity. So currently there are some studies, quite a few vaccines which have been evaluated in non-human primates. And in fact, the natural infection in non-human primates, when people have tried to previously infected non-human primates, there has been data to show that they are protected from reinfection. Okay, so this is a proof of principle that immune response generated by the SARS-CoV-2 infection is able to protect the animals later on from a new exposure. So the natural infection induces both mucosal antibody responses. For example, the upper respiratory tract, where you have the mucosa, as well as systemic antibody responses. So the mucosal antibody responses are mediated mainly by the secretory IgA, and the systemic antibodies are mainly mediated by the IgG. The upper respiratory tract is protected by secretory IgA, as I told you, and the lower respiratory tract is protected by IgG. So intramuscularly or intradermally injected vaccines induce mostly IgG, but no secretory IgA. So I would like to emphasize that the route of immunization, with respect to viruses, to protect against viruses which cause respiratory infection, for example, is extremely important. It is therefore possible that most vaccines currently in development induce disease-preventing or disease-attenuating immunity, but not necessarily sterilizing immunity. What I mean to say is, if you need sterilizing immunity, the route of infection, that is upper respiratory tract, also needs to have a protective immune response, which is mediated by secretory IgA. So, and what are the different vaccine designs that are being approached for SARS-CoV-2 vaccine development? So this is the SARS-CoV-2 virus, the whole virus with its genome, the nucleoprotein, the envelope protein here, and the S protein, the spike protein. So if you look at one of the primary antigenic targets, that is a spike protein, and most vaccines, whatever the design is based on expression of the spike protein or just the receptor binding domain of the spike protein. So the various examples of different kinds of vaccines are, you can have just the SARS-CoV-2 vaccine strain which has been inactivated. So the vaccine manufacturers grow the virus in bulk, in huge volumes, and they're inactivated chemically, and their safety is evaluated. So in fact, there are at least a few vaccines which are progressing through to phase 3 clinical trials, which are inactivated vaccines. And then you can have live attenuated vaccines that are basically the original version of the virus, which has been weakened genetically so that it just generates immune response, but you don't have disease being caused by this particular vaccine. So there are not many encouraging candidates in this particular section, that is live attenuated vaccines at this point. Okay, so most of the vaccines that we have in under different phases of clinical trials are based on this design, the recombinant vaccines. So earlier speakers also mentioned to you that the recombinant spike protein based vaccines and the recombinant RBD protein based vaccines are the most commonly developed kind of vaccines for SARS-CoV-2. And you also have some inactivated vector vaccines that carry copies of the spike on their surface but have been chemically inactivated. So there are at least 2 vaccines in this category, which is one from Russia and one from China, basically, which are progressing into clinical trials. And you also have other kinds of replication competent vector vaccines. So basically a different vector, a different virus, basically the adenovirus, for example, which is a very commonly used virus to deliver vaccine antigens of other viruses. So that is another category. So it can be a replicating one, and you may also have a non-replicating competent vector vaccine, which again expresses the S protein. And another group of vaccines, the nucleic acid vaccines, nucleic acid based vaccines, are very appealing kind of vaccine approaches. And we have, in fact, one of the vaccines in India being progress to clinical trials is a DNA vaccine against SARS-CoV-2. And another very important approach based on RNA vaccine, mRNA vaccine, which has again progress, which is making fast progress, and it might be available at least in the Western countries soon, is based on RNA vaccine approach. And you can also have some virus-like particles, or the VLPs, which are not, they're not complete viral particles. They lack the genome, but they express the antigenic targets, that is the spike protein in this case, on the surface, and they're also shown to be very efficient vaccines. And what do these vaccines primarily try to do? Whatever category the vaccines belong to, whether it's inactivated or expressed by a virus vector, whether it's a subunit vaccine or a virus-like particle, or whether it's a DNA vaccine or a RNA vaccine. So all these different vaccines are delivered into the host cell, wherein the host APC, or the antigen presenting cell, will take up these vaccines, and antigens are expressed and presented to the T cells, basically the CD4 T cell and the CD8 T cell, or the B cell. Now, in the case of B cells, they get activated and start proliferating, and they start secreting antibodies. And these antibodies are neutralizing in nature. They can block the attachment of the SARS-CoV-2 virus to the host cells. And in case of CD4 T cells, their primary function is to help B cells. The B cells and the CD8 T cells as well. So they secrete a lot of cytokines to bring about their helper function. And some of these activated CD4 T cells and activated B cells differentiate into long-term surviving memory cells. Similarly, with the CD8 T cells, they get activated, and they are equipped with certain proteins which induce infection, which induce lysis of infected cells. So these are the different ways they can protect a vaccinated individual. The CD8 T cells can lyse infected cells. CD4 T cells help B cells and T cells to do their effector functions. B cells produce antibodies. So whatever the nature of the vaccine is, the whole idea is to induce these protective immune responses. And in fact, I was telling you, the SARS-CoV-1 helped us learn a lot about the S protein, how S protein can be a candidate vaccine antigen. And people, in fact, have contemplated on using a SARS-CoV-1 vaccine to protect against SARS-CoV-2, where they show that you see both these viruses target the ACE2. So if you have antibodies against the S protein of SARS-CoV-1, can it protect against SARS-CoV-2? So people have tried to look at that, and they have found some encouraging results. In fact, which means some of the initial problems of vaccine development can be progress fast without much hassle. So ideally, vaccine for COVID-19 should progress through all these different stages, and wherein you have the initial targets being evaluated, and small batches of vaccines are prepared, in vitro tested for their safety and proliferation or toxicity profiles, and then the targets are validated and their immunogenicity is evaluated, and further efficacy and protection studies are done in human animal models, and progress to, if all these different stages give encouraging results, then it progresses to phase 1 human clinical trials and phase 2 clinical trials in all quick succession, and finally to phase 3. So you have a successful ideal COVID-19 vaccine with manufacturing capabilities and well standardized supply chain, so that there is enough distribution across the world. Of course, some of the disadvantages could be some of the side effects, adverse side effects. In fact, vaccine-induced enhancement of the disease is one important concern that people have encountered with the SARS-CoV-1 vaccine or the MERS-CoV vaccine. So people are trying to look at whether SARS-CoV-2 vaccines, before being administered into the humans, they need to be evaluated for these side effects. And of course, lack of efficacy, if there's minimal or no protection at the end stage, then it's a waste of huge amounts of effort. When you look at different categories of the vaccines, what are the advantages and the disadvantages? I'm not going to read the whole list, just quickly. So the DNA vaccines or the RNA vaccine, you don't have handling of infectious material, as in like when you're dealing with the inactivated virus, for example, or a live attenuated virus, you will have to deal with live viruses. But with the DNA and RNA vaccine, you don't have to do that. So which means they can be also used in immunocompromised subjects. There is no danger of these live viruses causing disease, and they have been shown to induce reasonable and enduring immune response, both humoral, that is antibody mediated, and T cell mediated immune response. So those are some of the advantages. What are the disadvantages? Until now, there are no approved DNA vaccines for human usage. So we don't have a real world experience in the global population of people having received DNA vaccines and what is the safety profile, efficacy profile of these vaccines. And some people have looked and seen that DNA vaccines can cause variable mucosal immunity and other immune responses, which means they're not like very uniform with respect to inducing these immune responses. And some require special tools for delivery to subdermal layer in the skin and potential risk for host cell genomic integration. So that might lead to other issues and other ethical issues that which might block the progress of the vaccine development. And with respect to RNA vaccines, they can be rapidly designed and produced. Again, there's no handling of infectious material, no potential for insertional mutagenesis, and there people have demonstrated strong early antiviral responses in both compartments. Okay, again, we don't have any approved RNA vaccines that have been in human usage. And with respect to protein vaccines, we don't have infectious material, strong antibody responses, but they need adjuvants. So these are some of the common approaches wherein these vaccines have a lot of advantages, but also some disadvantages. But these are all not unsurmountable. We can all overcome these and make sure that we have immunogenic vaccines. And what are the evolving scenarios for global COVID-19 vaccine development at this stage? So if you have a vaccine, so you can have two types of vaccines actually now. The pace at which we are going, you can have a pandemic vaccine, which is not really thoroughly investigated at all, in all different stages, like what we are seeing in China and Russia. Some vaccines which have not gone through clinical phase 3 clinical trials are already approved for usage in the clinic. So you may have a vaccine like that, or you may have a fully validated, rationalized vaccine pipeline. So whatever the vaccines are, so the priority vaccination groups would be healthcare workers, individuals with comorbidities, senior citizens, and ethnic minorities. And after that, the other groups that can be considered for vaccination are people who have been exposed to the SARS-CoV-2 infection, but the immunity is waning. So they may need to be given a vaccine. And people who have got the pandemic vaccine, but the immunity is waning because that vaccine is not shown to be highly efficacious. So they may be the second set of individuals who may need the vaccine. And you can have mass vaccination, that is the last in the list, wherein you have vaccines at the national level, continental level, and the global level. So the best vaccine strategies identified from the rationalized vaccine paradigm will be pushed to get into mass vaccination for worldwide immunization. So as we saw earlier, a pandemic vaccine paradigm is like 1 to 2 years, which might actually escape many stringent guidelines for vaccine production, versus a rationalized vaccine paradigm, which will take 10 to 15 years. As I mentioned earlier, so the kind of the vaccine decides how well we are protected in the upper respiratory tract and the lower respiratory tract in case of SARS-CoV-2 vaccines. So mucosal and systemic immune responses to natural infection with respiratory viruses and vaccination. So when the exposure happens through natural infection, so you have, so this green oval colored the area shows you the upper respiratory tract, and here the this represents the lower respiratory tract. So through natural infection, you have both the upper and lower respiratory tracts being protected, like their protective immune responses are generated. And the upper respiratory tract, as I said earlier, is mostly through the secretory IgA1 antibody, and the lower respiratory tract, that is a systemic immune response, is through mostly IgG1 antibody. So if somebody is administered intramuscular or intradermal vaccination, then they're unlikely to develop upper respiratory tract immune response. There is no secretory IgA, but they may have a good systemic immune response in the lower respiratory tract. If you have intranasal vaccination, as against intramuscular or intradermal, then you can have both upper respiratory tract and the lower respiratory tract with protective immune responses. Okay, so those are certain parameters for considering to be considered during vaccine administration. So when you look at various vaccine candidates, they may also have potential adverse effects and many benefits, of course. So with respect to whether it's inactivated, virus vector, or the S protein subunit vaccine, or the DNA vaccine, or the RNA vaccine, as I said, so there might be a risk of antibody-dependent enhancement, vaccine-mediated enhancement of the disease on lung immunopathology. And of course, the most important advantages or benefits of the vaccine are induction of neutralizing antibodies, T-cell responses, and antibody-dependent cellular cytotoxicity and other modes of protection. There is decreased morbidity and mortality. So when you look at the global vaccine landscape, so these are all the different categories: recombinant viral vector vaccines, about 42 of them, out of which majority of them are in the preclinical development. About 4 have progressed into clinical development. And among nucleic acid based vaccines, about 10 has progressed to clinical development. And we have very few candidates of live attenuated vaccines. And so virus-like particles also form a major part of vaccine development efforts. Okay, so all these different vaccines, so about 30 of them have moved into different phases of clinical trials. So this is just another pictorial representation of how different categories of vaccines. Here it represents an inactivated vaccine, and this is a live attenuated vaccine. So we don't have any live attenuated vaccine in any of the preclinical or the clinical phase trials that is being evaluated. But you have a lot of recombinant spike protein based vaccines and RBD based vaccines, which are going through phase 1, phase 2, and progressing towards phase 3. So you have some inactivated vaccines which are under evaluation under phase 3 clinical trials, and you have non-replication competent vector vaccines, which some of them are in phase 3, and in fact 2 of them have been licensed for human usage, which I mentioned earlier. These licensed vaccines, which belong to this non-replication competent vector vaccines, were the CanSino vaccine, which is used in the Chinese military, and the vaccine by Gamaleya Research Institute in Russia, which is, both of these were licensed. Both of these were licensed without a phase 3 clinical trial. And so this picture represents 14 different vaccines, again, and all these different categories: nucleic acid based vaccines, proteins, viral vectors, all these in different stages of development. And quite a few of them are hoping to be available by somewhere along 2021, somewhere in the beginning. Some vaccines are, the adenovirus based vaccine here is hoping to be available at in early 2021. And quite a few vaccines, like the one which is in India, this particular vaccine, about 400 million doses are expected to be available by the end of this year. So, so in preclinical trials, when people looked at immune responses to different vaccines, like the Sinovac and all these different companies which are producing vaccines based on different strategies, whether it's an inactivated vaccine or adenoviral non-replicating vaccine here, or an mRNA vaccine, or a subunit vaccine. So they have looked at antibody, neutralizing antibody responses in many of these cases. So here they have not demonstrated any neutralizing antibody titer, but quite a few of them have, at least 3 of them have shown neutralizing antibody titers in people who have been administered this vaccine, and this has been shown to increase. The neutralizing antibody titer has been shown to increase after boost. Here they did not find anything, but here after boost they're able to find. Here it's enhanced. Again, here it's the antibody titers are enhanced in all these different cases. And also T cell responses have been shown by at least 3 of these different vaccines, basically the adenoviral vector vaccine and the mRNA vaccine. So just to say that in the preclinical stage, they did see evidence for these vaccines being immunogenic in nature. And most of these studies have been carried out in these rhesus macaques or cynomolgus macaques. Okay. And these are data from phase 1 and phase 2 studies in human clinical trials. Again, they have seen after boosting that antibody titers are enhanced with these different categories of vaccines, and T cells are also shown to be induced or activated with these different vaccines here. Especially with the mRNA vaccine, there's been good CD4 T cell response and low CD8 T cell response. So I would like to just skip a few slides. I think I'm gone beyond my time.
The speaker is waiting, sir. Please.
Sorry. So I'll just try to skip the. This is just evidence from mRNA vaccine to show that you have good CD4 T cell responses being induced. And so the mRNA vaccine 1273 has been shown to be a very robust vaccine. And another appealing technology is a nanoparticle platform vaccine technology, wherein it's very easy to administer the vaccine antigens coated on nanoparticles. And you have India COVID-19 vaccine update. For example, we have at least 3 vaccines which were mentioned earlier. Covaxins, that is from Bharat Biotech, and one from Zydus Cadila, and one in collaboration with Oxford University and AstraZeneca with Serum Institute. So they're all in different phases of clinical trials. So with that, I thank you all for patient listening. I apologize for going over time.
So it's a very wonderful. It's a very wonderful information on the vaccine, the present scenario, and the global understanding of the vaccine. Here we have a question.
Yes.
Recently, that South Africa started some trial using tubercular vaccine, the BCG, against COVID-19. You please, how can you just do it? Because
yeah, I just had a slide on that. Sorry. Yeah, so this is something called trained immunity. So the BCG vaccination endows our cells, monocytes basically, with characteristics of trained immunity through epigenetic and metabolic rewiring of myeloid progenitors in the bone marrow, which means to say there might be some non-specific cross-protective responses induced by this particular vaccine, BCG vaccine. So that is why people are trying to look at protective efficacy of this vaccine. It has in fact been shown to be helpful in other infections as well. So, but there is no clear data as to how well it does with protection against SARS-CoV-2.
Thank you
Does
for
that answer my question? Answer your question? Sorry.
Yeah, thank you. Thank you for joining with us, Dr. Swami.
Thank you, sir. Thank you again. Thanks for.
We have the last speaker of today's session, Dr. Claudia Perkel. She's Dr. Phil Claudia Perkel, basically from an Oriental background of studies from the Ruhr University, Bochum, Germany. And now her area of interest are very wonderful because she worked on the Oriental studies, and she developed a lot of interest in understanding Unani system of medicine and the Mughal culture, development of Unani system of medicine in Pan-India. She did a lot of work as exploratory findings are there from the Unani prospect point of view. Based on her interest and some of the things, we requested Dr. Claudia to come and to speak something about the topic. We have shared her to discuss about the historical perspective of infectious disease in Unani system of medicine. Dr. Claudia is an intercultural trainer. She's a scholar, a blogger on particularly the Mughal history and Unani medicine. With this brief introduction, I request Request Dr. Claudia. It's over to Claudia
Yes.
for the presentation. Thank you.
Thank you very much for your very kind invitation first, and then for the very kind introduction of my biography. And indeed, as you might see from my background, my Zoom background today, I chose to show you some pieces of literature on the infectious disease. And the first thing I have chosen, the first book, is one you have already mentioned in your introductory remarks, but I will tell you further. And the second one is from the Mughal history. And so let's go on. As you already said in your introduction, infectious diseases in Unani literature and Unani or India, I might say, medical literature, are the measles, Hasba, the pest, Taun, cholera, Heiser, and smallpox, Judari. These are the most common diseases mentioned there. One of the most important works in this regard, of course, is Arazi, the author and the famous, I would say, universal scholar who published extensively on medicine also. And he died in 925. And he wrote this very informative book, Kitab al-Judari wal-Hasba. And I would like to say, I consulted the Arabic book from the University of Beirut. But one has to say, of course, in the meantime, since it has been published first, it has been intensively translated also into English and German, French, because Rhazes, as he is known in European medical history, is a very famous scholar in our culture also. So let's go into the detail. He says that the measles belong to the category of Amraz Awaiya. And he says, as for old men, smallpox seldom happens, except in pestilential, putrid, and malignant air. And he adds, putrid air has an undue proportion of heat and moisture. And he directly says that the Asbabasita azaruria are closely connected to this type of diseases, to the Amraz Awaiya. As Hawa, of course, is the first of them. Here, the next excerpt of the book. The bodies most disposed to the smallpox are in general such are moist, pale, and fleshy, the well-colored also and ruddy, as likewise the swarthy, and when they are loaded with flesh. Those who are frequently attacked by acute and continued fevers, bleeding of the nose, inflammation of the eyes, and white and red pustules and vesicles. Those that are very fond of sweet things, especially dates, honey, figs, and grapes, and all those kinds of sweets in which there are thick and dense substance. I think, why did I choose this text? I think it's very important to say that Razi, maybe like no other except Ibn Sina, differentiates between the different kinds of temperaments affected by diseases. Meaning, as I always say, Dr. Alimuddin's measles or smallpox might be very different from my smallpox or measles, and it really depends on the temperament of the patient, and this is quite important to say. Again, continued, bodies are lean, bilious, hot, and dry, are more disposed to the measles than to the smallpox. Those bodies that are lean and dry and of a cold temperament are neither disposed to smallpox nor to the measles. I think this is also very, so it is very important to differentiate between the temperaments. And I think this is of course the strength of the Unani system, that it differentiates between the temperaments. And I don't know, I have to leave this question to the medical experts, if it's also in the case with COVID-19, that the different temperaments of the patients you are more likely to exposed to COVID-19 or not. I leave it open and maybe you discuss this point later. Okay, what we have seen in the first text is that this is not only connected to the Asbabasita, but to the theory of the miasma, the bad air, which has its origins in medical teachings of ancient Greece. Disease is caused by polluted air or contaminated winds carrying particles of rotten material. And I think it's this theory is culturally very effective in Europe, as you can see it in the picture, which is a 19th century picture on the spread of cholera by miasma. And also it is very important in our culture, for example, ventilating rooms and closing the shutters at night and in the evening due to the fear of miasma. And one might say miasmatic theories are contradictory to Unani, but I think it has, due to the Asbabasita Zaruhia, it could be integrated in the Unani perspective. A second theory, of course, which was integrated, one might say, in Unani theory was the theory of bacteria. This is also 19th century, from a 19th century Unani manuscript, which is in my collection. So it is very important to go back to the theory of bacteria to the discovery. And bacteria were discovered by Anthony van Leeuwenhoek, who was a merchant from the Netherlands, who discovered the bacteria at first in 1675, which of course was really, he did not intend it, but it took 200 years until the research of Robert Koch, Ignaz Semmelweis, and Louis Pasteur, sorry for the slip, finally proved the existence of bacteria. And later it was very important that all the theories, miasmatic theory, bacteria theory, were discussed during the outbreak of the cholera in London in 1845. But concerning the pest, the pathogen Yersinia pestis was only discovered in 1894. At the same time, Yersin, who discovered the Yersinia pestis, which is named after him, he discovered that there was a connection between the outbreak of the pest in Singapore and the death of the complete local rat population at the same time, so giving a proof to the theory of zoonosis. Yeah, so now we are going back to the Indian literature, and one of the my most favorite piece of literature at all. It is written by Nuruddin Jahangir, and it is his autobiography, Suzuki Jahangiri, today popularly known as Jahangirnama. Jahangir was the son of Jalaluddin Akbar, the famous Mughal ruler, and he himself became his successor and ruled Hindustan from 1605 till 1627. And what makes him so interesting, it is his great interest in biology, the fauna and flora and the medicine. And I'm showing some examples of his autobiography, and I hope it might become clear why I'm so in love with this text. He said about the plague On the first day, a headache and fever occur, and much blood flows from the nose. On the second day, they die, meaning the patients. In any house where one has died, all the inhabitants are certain to be lost. Everyone who comes near a sick person or corpse is similarly stricken. There was one person who had died, and they laid his body on straw to wash it. By chance, a cow ate some of the straw and died. Thereupon, several dogs ate some of the cow's flesh, and they all died. Things have gotten so bad that neither will fathers come near their sons for fear or death, nor will sons approach their fathers. Of course, here the principle of zoonosis is wonderfully explained. Of course, Jahangir had never heard about this conception of this, but he observed it without knowing the medical details. Second, and this is the last highlighted sentence, it is shown that the system of quarantine was also observed there. Because fathers will not come near their sons and vice versa. Second text. During this year, and indeed during the 10th year, a great pestilence appears in several places in Hindustan. The epidemic began in the Punjab countryside and spread little by little to the city of Lahore. Many people, both Muslims and Hindus, died of disease. After that, it reached their hand and so on, continuing about this, that this disease is so dangerous, it afflicts the whole population. And he brings, of course, back the religious situation of Hindustan back into the literature, but this is only a way. Okay. So again, since it appears from what aged people say and say from history books that this illness has never appeared in this province before, the physicians and learned were asked about this cause. Some say that since there have been 2 successive dry years with little rain during the monsoon, it has been caused by the putridity of the air arising from the dryness and lack of rain. Others attribute it to other causes. God only knows, one can only bow to divine destiny. What is a slave who will not bend his neck to orders to do? Here, I think this text is interesting because it is clear that Jahangir fully knew about the Unani principles of medicine because he always consulted the physicians and the learned, meaning the hukama. And it is also coming back the putridity of the air, meaning the miasma theory I already talked about. And what is interesting, it is that water, which is not part of the Asbabesitta Zawriya, is coming into this theory of medicine also, especially in the Indian context. And I think this is quite interesting because India has a monsoon, Europe has not. So the question of water might not be that interesting in the Indian context before. So again, concerning the water thing, there is a river in Rajour, the water of which becomes extremely poisonous during the monsoon season. Most of the people get a swelling on their necks and turn yellow and weak. I think here again, the question of poisonous water is back into discussion, meaning that Jahangir knew about this thing. And one of my favorite excerpts is here. The late Asaf Khan's daughter, Asaf Khan, by the way, was the brother of Nur Jahan, Jahangir's famous wife, who is in the house of Khan Azam's son Abdullah, told me a very strange story and insisted that it was true. I found it so strange that I record it here. She said, one day I saw a mouse in the courtyard of the house. It was staggering and reeling this way, and that I was though it were drunk. It was going in all directions and didn't know where it was going. I told one of the maidservants to catch it by the tail and throw it to the cat. The cat leapt up in delight and took the mouse in its mouth, but it put it down at once and made its distaste obvious. little by little, signs of distress and suffering came across its face. Okay, so we can see the infection between the animals. Continued, the next day it was nearly dead. It occurred to me that it should be dosed with a little antidote. When its mouth was forced open, the inside of its mouth and its tongue looked black. It was in a terrible state for 3 days, but on the 4th day it came to, after a maidservant got a plague bubo. She had such distress and pain that there was no consoling her. Her color too changed to blackish yellow. Then she got a raging fever, and the next day she had uncontrollable vomiting and diarrhea and died. 7 or 8 people in the house died in the same manner. Here again, the way of a zoonosis can be perfectly shown. Again, and I want to say, this is the text from the 17th century. And of course, Jahangir has never heard about all these things of infection and the plague. So what are, so to speak, the results of my talk? I want to say that for gaining medical information, it might be also very useful to consult different sources from medical sources. So I think it's very important to see biographies and autobiographies to get medical information, which maybe we one day might also have in the case of COVID-19. So maybe one day we will look at the literature from different parts of the world. It might become clear that the way of the infection was different, or we can find a solution to heal a disease. So I think there should be a quite open approach to literature and literature study. Also, I think the weight of even the Mughals to speak of zoonosis is very interesting, and I think humanity has to check how we want to live together with the animals and nature in future to prevent diseases like this. This is, I think, also very important. And third, going back to the first source I showed, meaning, as I said, the strength of Unani system is, of course, its individual approach and the analysis of people's temperament. So I think this might, or this should also be included in that approach. So these are, of course, some remarks in ongoing research and questions I do not only ask myself, but also might ask you. And I want to thank you to give me the opportunity for the presentation. Thank you very much.
बहुत शुक्रिया Dr. Claudia ji. बहुत शुक्रिया. Thank you. You are giving the wonderful reference of historical perspective of this infectious disease, particularly with reference to the Mughal kingdom, Mughal ruler Jahangir Nama, whose era was between 1605 to 1626. It is a really wonderful collection. Unani system not only speaks about the disequilibrium among the body fluids, but also they have a concept of microorganism also. But the point what you have raised here, this is the time where the Leeuwenhoek was not developed any microorganism visible instrument. So it was the time only after the period of the Renaissance, it came to know that there are organisms. Germ theory came into existence to understand the diseases. But the wisdom of the Unani is there. It evidences the concept of infectious diseases are there. They have well educated, aware about the prevention, precautionary measures to be taken during the infection. Not only that, but also the therapeutic part of the infectious disease also dealt in detail. Thank you for joining with us. If you have a time, please tomorrow join with us also. And with this, we have come to the conclusion for the day first session. I extend my thanks on behalf of the organizing committee and the director of this institution to all the panelists, starting from Professor V. Ravi sir, for his wonderful deliberation on the virology. And next, Dr. Shashibhushan sir on clinical manifestations of COVID-19, particularly, and my thanks to Dr. Giridhar Babu sir on epidemiology of this COVID-19, and my thanks to Dr. Manjunath, who gave an elaborate description on the current status of the vaccine and different forms of the vaccine. And my thanks to Dr. Claudia ji once again. Bahut bahut shukriya, Dr. Claudia ji, for joining with us. Now today we are concluding this session. Tomorrow we will be joining with a few more international personalities of international repute in Unani system and allied sciences. Dr. Kamil from United Arab Emirates and Dr. Professor Ziaur Rahman from the India's most prestigious university, Aligarh Muslim University. Next followed by, we have a panelist from Indian Institute of Indigenous Medicine, Colombo, Dr. Fazina Hamid, and we have Dr. Mariam Khat from Kasan, Iran. Madam is with us since morning. Thank you, madam, for joining with us. And Dr. Nazrul Islam from Bangladesh of Hamdard University. We hope so. Tomorrow again, we are going to meet by sharp 11 o'clock. Till that, thank you. Thank you for all joining with us, all panelists and participants. Inshallah.